Hi, hello. Hi, everyone. Audible, visible? Hi, students. Am I audible and visible to you all? Hi, everyone. Am I audible and visible to you all? A very good evening. Let me refresh. I hope I'm clearly audible and visible to you all. Hi, so very good evening everyone. Welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma. So I hope you all are doing very well. So see, we are moving as per your demand. Ma'am, structural organization in animals. Ma'am has started that. Ma'am, anatomy of flowering plants. Ma'am has started that. Ma'am, please go for the human physiology first. I'm starting that too. So I hope you guys I hope you people have checked the channel. I hope you people have checked the thumbnails. The next chapter that I'm going to start is your digestion and absorption. But before that, I will teach you about the enzymes. Okay, the classification of enzymes as well. So ma'am is, right, keeping her promises. But my students are not keeping their promises. Isn't it? I'm sorry for being late. I was starting the system. But as usual, you know that. Right? Right? That in my case, all the troubles, they are waiting for me. They are like, ki chalo, Ambika ma'am ki class hai, we'll go there. Hai na? So, something messed up. So, that is why I started late. But at the end, like we are starting the session, we are going to start the concepts, and today it is actually the one shot. So in today's lecture, we will talk about the tissues, about the meristematic tissue, permanent tissue. We will even discuss about the ground tissues, how the distribution is there, about the sections as well, and then we will say tata bye bye to each other. And yes, there is one class on Sunday as well, because there is one class on Sunday as well, and Sunday at 1 p.m. I'm going to take your session. That session will hardly take 1 hour or 2 hours. Oh, sorry, hardly take 1 hour or 1.5 hour. So in that session, we are going to discuss what? We are going to discuss secondary growth. Because for secondary growth, I need your full attention. That is why I am taking it separately. Clear? 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 Okay. Acha, you know something amazing? Do you know something amazing? I think you know that. And do you know what is that something amazing? That something amazing is our HSP sir's batch. Okay, right? Many students were saying that, "Ore, HSP sir, he's amazing. His tricks are amazing. He's wonderful, right? He can make anything easy." So he said, why not I should so literally it was HSP sir's idea that I should start a batch. Okay, I will make chemistry easy for them. All the time they cry for the chemistry. Chalo, let's make it easy for them. So he has started one batch, and that is Chemistry Mind Maps by HSP sir. And let me tell you, students, right, just because he's the part of my team, that is not the reason I appreciate him. But actually, let me tell you, bache, students, they used to pay in lakhs to attend his classes. Okay, literally, students used to pay in lakhs to attend his classes, right? From last 3 years, he's teaching online, but before that, he was there in the in a very renowned offline center, in the very renowned offline institution, and students, they just love him. Okay, they just love him, and you are going to get his course in just rupees 2,000. Just rupees 2,000. Just imagine. Okay. And I am not at all bragging. Let me tell you this thing honestly, right? He's teaching from last 25 to 27 years, and he was the part of a renowned offline institution, and where students, they were paying in lakhs just to get the badge of HSP sir, because when he teaches chemistry, then chemistry is crystal clear, then chemistry is the easiest subject ever. Okay. Okay, so join it. If you're the part of this batch, very good. Otherwise, join this batch. What do you have to do? It's very simple. Click on the link, add your number, add your email, and then next is what? You're going to get this batch at a price of 1999, right? So be the part of this batch. ASAP, be the part of this batch. And now, let me start the chapter. And yes, one more thing. These days, bache, I have started catering the issues that students used to face. Just say they don't know how to study, they don't know how to manage the time, right? They don't know how to prepare in a better way for the biology. So I have made the videos on them. Now my next video, right, you are going to get it tomorrow. Tomorrow, what will you get? There will be one video. In that video, I will tell you how to read the NCERT. Okay, how to read the NCERT. That is what I'm going to tell you. Do watch that video. Okay, all your concepts will be clear after watching that video. Concepts means you will get to know how to read the NCERT. Simple as that. Okay, so now let's start the chapter, and the chapter is anatomy. So first of all, tell me, what is the meaning of anatomy? Yes, the very first question that you need to answer is that what is the meaning of anatomy? What do you understand by this word anatomy? Yes, today you will get to know about many things. I'm going to share many things with you. I will share, obviously I will explain everything from the NCERT, right? We will make NCERT easy, but yes, few 1 to 2 points extra which were there in PYQs, which were actually the PYQs, that I am going to share with you all. So first of all, tell me, what do you understand by the word anatomy? Yes, what do you understand by the word anatomy? Study of internal structure. Exactly. Obviously, you are right. So, anatomy is the branch of biology or branch of botany that, because it is the anatomy of flowering plants. Okay. So, it is the branch of biology or branch of botany that deals with the study of internal structure, isn't it? And moreover, not just the internal structure, here it also deals with the organization of plant organ as well. Okay, so when it comes to the anatomy of the flowering plants, simple anatomy, study of internal structure, branch of biology that deals with the study of internal structure. But if I'm specifying that anatomy of flowering plants, then it is the branch of botany which deals with the study of the internal structure and the organization. Okay, and the organization of plant organs and structures. Okay, that is what anatomy is. Now in this chapter, how are we going to start? We are going to discuss the tissue first. Like, basically, you know that bache, all the living organisms, they are made up of cell. We all are made up of cell, है ना? We all are made up of cell. Yes or no? Yes or no? We all are made up of cell, because it is the cell which is the basic structural and functional unit of life. It is the fundamental unit of life. Below the level of cell, there is no independent existence of life, right? Below the level of cell, there is no life at all. Now, bache, group of cells, fine. Group of cells having similar or dissimilar function, having similar or dissimilar structure, but having common origin. That is very important. Right, a group of cells having similar or dissimilar structure, a group of cell having similar or dissimilar function but having similar origin, that are making the tissue. That are making what? That are making the tissue. You know this hierarchy, we have even discussed in structural organization in animal. So cell will form tissue, tissues are going to form the organ, many organs together will form the organ system, and finally what are we going to get? We will have the organism. Right, we will have the organism. This is what you need to keep in your mind. Clear, bache? Clear, bache? So what is a tissue? What is a tissue? Everyone just write it down in the chat section quickly. It's a group of cell having similar structure, having similar function, and having similar origin. In the last chapter, structural organization in animal, in the last chapter also I told you, bache, the structure can be dissimilar, the function can be dissimilar, but in the case of a tissue, the origin of cells should always be similar. Clear, bache? The origin of cells should always be similar. Is that clear? Yes or no? Is that clear? Okay, so group of cell having common origin usually perform a common origin. Let me write it here. So group of cell having common origin and usually performing common function. It is what a tissue is. Clear, bache? It is what a tissue is. Any doubt? Yes, bacho. Any doubt here? Yes. So how can we divide the tissue? We can divide the tissue into two groups, right? How can we divide it? We can divide the tissue. Array. Wow, even that's beautiful. So we can divide the tissue into two groups. So what are we going to study here, bache? Can you tell me? Can you tell me what are we going to study here? Right, we are going to divide the tissue into different groups. So first of all, right, on the basis of capability of division, the first criteria that we are going to set is, Subhashini, the first criteria that we are going to set is, right, on the basis of capability of the division. Let me write it here. So as I said, bache, on the basis of capability of division, same way we will see on the basis of on the basis of origin, on the basis of position also we divide the tissue. The very first thing that we need to understand is on the basis of capability of division. So here you have the meristematic tissue, right, bache, and then you have the permanent tissue. These are the two things that we are going to study here. One is the meristematic tissue, right, and another one is the permanent tissue. So when it is the meristematic tissue, right, actively dividing cells, or you can say that, let me write it here, bache, please make the notes, bache, actively dividing cells, right? Here what will you see? Actively dividing cells. So basically they are undifferentiated cells. What type of cells are they? They are undifferentiated cells. Undifferentiated means they are just dividing, they do not have a permanent function, right? They are just dividing, they do not have a permanent function. Such cells are meristematic cells, actively dividing cells, or you can say that undifferentiated cells. But when they are permanent, right, they are differentiated cells. So bache, when we use the word differentiated, okay, when we use the word differentiated cells, means they have acquired a permanent function. Just in the case of plants, simple, I'll give you one example. Root tips are having meristem, shoot tip is also having meristem. Now permanent, when it is the permanent tissue, when it is what? When it is the permanent tissue. So in the case of permanent tissue, what's the point? They are having a specific role, jaise parenchyma, collenchyma, sclerenchyma. I hope you all know about parenchyma, collenchyma, sclerenchyma. Even if you don't know, don't worry, today I will teach you, right? First of all, hit the like button, and if you're new to our channel, then hit the subscribe button as well, and share this video with your friends as well. Okay, we literally need your support, let me tell you, students. Okay, if we want to help you here, see, on YouTube we want to help those who cannot afford anything good, okay? So we want to help such students, and for that literally we need your support. Then only, you know, our organization will say ki, theek hai, okay, run your channel free of cost, it's fine, teach every day. Otherwise there will be a problem for us as well. So we literally need your support. So every time you join my sessions, share the link with others as well, other NEET aspirants as well, invite them to attend this class. Okay, chalo. So now focus here. Permanent means differentiated, jaise parenchyma in the case of plants. We have even started it in class 9th, I think. Okay, so these are the two things that we have. So on the basis of capability of division, this is what we have. Any doubt here, bache? Any doubt here? So can I not say that here I will be having immature cells? Immature means small in size and they are just dividing. Can I say so? They are the immature cells, they are just dividing. And here I can say that what am I going to get? I will get the mature cells here, right? I will get the mature cells here. So write down these points. So when it comes to the meristematic tissue, right, undifferentiated cells are there, immature cells are there, and let me tell you, bache, such cells are having large conspicuous nucleus, dense cytoplasm will be there, abundant plasmodesmatal connections will be there, very thin primary cell wall will be there. Okay, are you getting it? So no doubt it is undifferentiated, no doubt immature cells are there. So let me tell you, these cells are having thin wall. So thin wall means only primary wall is there, right? Dense cytoplasm you are going to find here. What will you find here? Dense cytoplasm. Conspicuous means visible nucleus is there, conspicuous nucleus. So thin wall, dense cytoplasm, conspicuous nucleus. These are the property of the cells of the meristematic tissue, right? And what will you see here, bache? You will see here in this tissue that there will be abundant plasmodesmatal connections, What are you going to see here? Abundant plasmodesmatal connections will be there. Abundant plasmodesmatal connections will be there. Are you getting my point? Are you getting my point? Right. That is what we discuss in the case of the meristematic tissue. Any doubt here? Any doubt here? So, bache, these points are important. They can ask you these points. Fine? Fine, fine, fine. Okay? Okay? Any doubt? Any doubt? And yes, thin wall, dense cytoplasm, conspicuous nucleus means large nucleus. It is one and the same thing, Srinath. And here, abundant plasmodesmatal connections will be there. And moreover, bache, when you talk about the meristematic tissue, na, here, it is a compact tissue. Do you know that? What type of tissue it is? It is a compact tissue. So, compact means intercellular spaces will not be there. So, intercellular spaces are absent. Okay. Intercellular spaces are absent. Are you getting it? Yes, intercellular spaces are what? They are absent. Clear, bache? Clear? Chalo, now I will ask you one question. Okay, that's a very simple question, of course. It's a very simple question. Now, meristematic cells, they are just dividing, and dividing. What are they doing? Dividing, and dividing. So, if they are just dividing, do you think that they will do photosynthesis? They will perform photosynthesis? Tell me. Very good, Ishwat. Excellent, bacha. Answer my question. I am saying that these meristematic cells, okay, these meristematic cells, they are just actively dividing. They are just dividing, and dividing. So, do you think they have the time for the photosynthesis? Of course not. So, in their case, this is also you can include in your notes that you will see that even the plastids are absent in these cells. Okay, even plastids are absent in these cells. And moreover, bache, right, vacuoles, if they are present, they are going to be very small, or you can say that they are absent. Okay, they are what? They are absent. Are you getting my point? So that is the point here. That is about the meristematic tissue. Are you getting it? So this is what you need to take care. So actively dividing cells, they are dense cytoplasm they have, right, bache? The large nucleus will be there. No non-living substances present. Only living substances are present. Clear, bache? Clear? And when it is the permanent, it is differentiated. These are the mature cells. They are performing a particular function. Okay, it is these cells are performing this tissue is performing a particular function. Are you getting my point? Yes, are you getting my point? Sure. Sure. So this is about the tissue. So now on the basis of capability of division, this is what we have, right? Now the next is on the basis of, yes, on the basis of origin. So can you tell me on the basis of origin, what do you have? Yes, Sunlight, Deena, Srinath, Shivakumar. On the basis of origin, what do we have? On the basis of origin and even on the basis of development. So see, bache, here we have three names. How many names do we have? We have three names here, right? First of all, we start from the promeristem. Let me explain it first, then I will make the notes. Okay, so first of all, it is promeristem, right? Promeristem means embryonic meristem. What type of meristem it is? It is embryonic meristem, right, which will form in the embryonic stages. Now this embryonic meristem, okay, now this embryonic meristem, it will form your primary meristem. What will it do? It will form your primary meristem. Okay, when these primary meristem, first of all, understand this part. I will explain it in detail. I will make the notes as well. Right now, it is just for your understanding. Okay, students, right now it is as per your understanding. Now, acha, tell me, are you excited for the human physiology? Tell me. Are you excited for the human physiology? Quick. Arey yaar, energy is not that good. Quick. Acha, plant physiology. Are you will get the plant physiology too. Big yes. Acha, you have completed Srinath. That's a very good thing. Acha, plant physiology also I will start. That should be the energy throughout the session. Okay. So pro-meristem, as I said, it is the embryonic meristem. Right, in the embryonic stages, this is what you will get, and then it comes the primary meristem, right? So primary meristem, right, meristematic cells, they will form some, obviously they are going to form some permanent tissues as well, and when that permanent tissues de-differentiated, when that permanent tissues they de-differentiated, right, and then you will get the secondary meristem. Now I know you must be thinking, especially those who are studying this for this first time, that ma'am, what is the meaning of de-differentiated, what is the meaning of this primary and permanent? This is what we are going to, this is what I'm going to explain now. So how are we going to start our process? How are we going to start everything? With the help of this pro-meristem, which is basically in the embryonic stages. Are you getting it? Are you getting it? It is basically from the embryonic stages. So ultimately this is the complete process. Okay, this is the complete process. Are you getting it? Now, as I said, pro-meristem, embryonic meristem. Okay, embryonic meristem, it forms in embryonic stages. It forms in embryonic stages. Now, what do you have to study under this title? So, this is what you need to focus majorly. Primary meristem and the secondary meristem. So, when it is the primary meristem, develops from, of course, बच्चे embryonic meristem will form primary meristem. जैसे suppose करो zygote है, zygote is dividing, embryo, fetus, then there is the human being, right? Same is the case here. If I am saying embryonic meristem, of course that embryonic meristem is going to form the primary meristem. So can I say that primary meristem, it develops from embryonic meristem? Can I say that it develops from embryonic meristem? Yes or no? And it helps in the primary growth of the body. It helps in primary growth of body. Now, what is the meaning of primary growth? So, primary growth of body means, yes, it helps in the primary growth of body. So, see, primary meristem we have, it develops from the embryonic meristem, it helps in the primary growth of body. So, primary growth of body means it helps in increasing the length of the body. Okay, the primary structure of the body will grow. This is suppose karo, if this is a plant, okay, if this is a plant, so its length will increase as per this. Okay, so it helps in increase in that length, right? It helps in increase in the length. Clear? Clear? Yes or no? Is it clear? So it develops from the embryonic meristem, helps in the primary growth of the body. So this is what primary meristem is. Clear, bache? This is what primary meristem is. Now, I will explain few things to you. Just say, let's start with the zygote. What is a zygote? Tell me. What is a zygote? Tell me, quick. Arey, please revert na. Quick. Quick. I'll tell you examples as well, but as of now, you answer my question. What is a zygote? One-celled fusion of male and basically the result of fertilization. The result of fertilization it is. Male and the female gamete will fuse. Male and the female gamete, after fusion, what will they form? They are going to form the zygote. They are going to form the zygote, and we know that zygote is diploid. And but when it comes to the zygote, we use one thing, and what is that? Totipotency. Totipotent. Zygote is what? Zygote is totipotent. Zygote is what? Zygote is totipotent. Now, what is the meaning of being totipotent? Right? That it is having the ability, it is able to develop into a complete organism. Right? It is able to develop into a complete organism. So whenever we say that a cell is totipotent, okay, totipotency is the property. Just a minute. Totipotency is the property. Okay, what is it? Totipotency. So totipotent means a cell is capable of growing into a complete organism. A cell is capable of growing into a complete organism. Are you getting my point? So this is what the totipotency is. So now we know that our zygote is what? Zygote is what? Our zygote is totipotent. It can grow, divide into, will form multicellular organism. Then there will be the complete body, this and that. This is what we know. But actually, how things are going on? Initially, there will be a stage when there is the active cell division only. Only the division is going on. Fine. Only the division is going on. Point? Yes or no? Guys, yes or no? Initially, the single cell has, if the single cell needs to divide, if it needs to form a complete organism, so obviously it will divide. It will divide. Same is the case here in the plants. So meristematic cell, they will just increase the cell number. They will just divide and divide. So they are capable of division initially. Right? Capable of division initially. These meristematic cell. But soon what is going to happen? These cells which are showing active cell division, they will undergo differentiation. Now, what is the meaning of differentiation, ma'am? Tell us this, hai na? We know this word, but what is the meaning of this word? Differentiation. So a single cell is dividing. A single cell is, the cells are dividing. They are meristematic. Now, that cells will undergo differentiation means now they will become mature. Now they will acquire a permanent function. What is happening here? What is happening here? This cell is undergoing differentiation. So differentiation means now it is attaining a permanent role. It is becoming mature. So it is the process of development actually. Right? This is the part of development. Whenever we talk about the development, we consider two things: growth plus differentiation. What we consider? We consider two things. We consider growth plus differentiation when we talk about the development. So during that development, a cell is going to increase its size as well, and then there will be the elongation of that cell as well, and it will undergo differentiation, and finally cell will become mature. Are you getting my point? Finally cell will become mature. So when it is the differentiation, so after that cell will become mature. After that cell become mature. Okay, what is going to happen after this differentiation? Cell become mature. It will acquire a permanent function. It acquire a permanent function. Are you getting my point? It acquires a permanent function. Okay. Okay, so here we have one cell. This cell is undergoing differentiation. I told you, first of all, that size will increase. The cell elongation will be there. Then finally differentiation will be there. And finally the cell will become mature. It is the part of development. It is the part of development. So this is what differentiation is. So you know, how do I explain it always? Always I explain it in this way. Just say now you are studying. You all are studying. So it is your cell division wala phase. You people are studying. It is your cell division wala phase. Right. Just in the case of cell, there is a division. You people are studying. Now, after studying, you become doctor. Right. Matlab, you are differentiated now. Right. It means you are differentiated now. You were studying. Now you are a doctor. You are differentiated. You acquire a permanent role. Right. You have acquired a permanent role. Now, again, you have decided that no, I'm not liking my profession. I want to do something else. Okay, I want to do something else. I'm not liking my profession. I want to do something else. Or I love my profession, but I want to do something else. It can be anything. Then what is going to happen? The doctor will start studying again. Means initially, right, you were studying and then you became doctor. So when you were studying, you were in that division phase. Now when you acquired a permanent role, that is now when you are a doctor, means you are differentiated. Now again you have decided that again you are going to study. right? So this is a de-differentiation. This is a de-differentiation. What is it? It is a de-differentiation. Okay, it is de-differentiation. Fine, fine. So now you are again studying, right? You are again de-differentiated. Means again, when we use the word de-differentiation, means a permanent cell or permanent tissue has again regained the capability to divide or the ability to divide. Okay, fine. So permanent tissue has again regained the ability to divide. That is the meaning of de-differentiation. Are you getting my point? That is the meaning of de-differentiation, that permanent tissue has again regained the ability to divide. Okay, and after that what is going to happen? Just now you are studying, let's say now you are an IAS officer, you are an IPS officer, or you are IFS officer, whatever it is. Okay, means again redifferentiation has occurred. So there are three words that you need to understand. Okay, there are three words that you need to understand. The first is differentiation. I hope the meaning of differentiation is clear to you now. Okay, then comes the de-differentiation. And then comes the redifferentiation. Okay, then comes what? Then it is the redifferentiation. Fine. So when meristematic cell, it means it is dividing. When it is differentiated, it has acquired a permanent role. Okay, now again when that permanent tissue, again when it undergoes de-differentiation, means regain, right? Means that tissue, that cell has regained the ability to divide. Is that clear? Yes, everyone. Is that clear? It has regained the ability to divide. And then again, once again, when it will acquire a permanent function, then it is redifferentiated. So that's how it works. Any doubt? Any doubt? If there is any doubt, do let me know, students, please. Right? Because this part is actually important for your NEET examination. Fine. Take it? Sure. So primary meristem mein, you have the example of apical meristem. You have the example of intercalary meristem. Done, bache? And even you have the example of intra. Its intra means within. Intrafascicular cambium. Intrafascicular cambium. That's the point that we have. Clear? That's the point that we have. Any doubt? Any doubt? Everything is given in NCERT. Trust me, everything is given in NCERT. Now, the second point is secondary meristem. I told you on the basis of origin and development. So first is pro-meristem, then primary, then secondary. Now so what is the meaning of secondary meristem? But secondary meristem is also known as lateral meristem. It is also known as cylindrical meristem. Do you know that? It is also known as lateral meristem. It is also known as cylindrical meristem. Clear? So when you talk about the secondary meristem, it forms from, or it develops from, the de-differentiation. This is the key word here, everyone. This is the key word here. It forms or it develops from the de-differentiation. Okay, from the de-differentiation of permanent tissue. Are you getting my point? By the de-differentiation of permanent tissue. Are you getting my point? Yes. It develops from the de-differentiation of permanent tissue. That's the keyword actually. There is a topic in NCERT, na? Secondary growth in the dicot stem. So even there we need to know about it. Okay. So it develops from the de-differentiation of the permanent tissue. So now you know that de-differentiation means the cells regain the ability to divide. They regain the ability to divide. Clear, clear, clear? Sure, everyone. Done? Secondary meristem. So secondary meristem, it helps in increase in diameter, or you can say that it helps in increase in diameter, or you can say that circumference, or in short you can say that it helps in increase in the girth. Okay, it helps in increase in the girth, so it helps in the widening of that particular plant. Okay, जैसे let's say see this is, let's say this part here, it is the apical meristem. So it is present, apical means something which is present on the at the tip. Take a stone, it is present at the tip. So this is the shoot apical meristem. Okay, shoot apical meristem, because it is present here. Take it, here you have the root apical meristem. Fine, root apical meristem, because it is present here at the root tip. Done. And here, fine, here you will see on the lateral sides, you will see that your secondary meristem is present. So it is formed by the dedifferentiation of the permanent tissue. So it helps in increasing the girth like this. It helps in increasing the girth. It helps in increasing the diameter, because it is present laterally, right? It is present laterally. That is why we also call it as lateral meristem and the cylindrical meristem. Done, bache? Any doubt here? Any doubt here in secondary meristem part? Tell me, any doubt here in secondary meristem part? Sure? Sure. So when it comes to the examples of the secondary meristem, which are again very important given in your NCERT, you have vascular cambium. You have vascular cambium. And let me tell you, mainly it is, I will explain it, bache, when I'll start the sections, especially interfascicular cambium. If you are aware of these terms, very good. If not, then don't worry, I'll tell you. Okay, so it is interfascicular cambium, vascular cambium, especially this interfascicular cambium. And other than that, cock cambium, which is also known as phallogen. It is also known as phallogen. So it is also the example of secondary meristem. Done, bache? It is also the example of secondary meristem. Done? Sure? Sure? Clear, bache? Okay, so that is what the secondary meristem is. So can we say that your primary meristem it will undergo, just a minute. So here you all can see primary meristem will undergo, yes, primary meristem will undergo differentiation, and what will we get? We will get primary permanent tissue. Now when this primary permanent tissue will undergo dedifferentiation, right, what are you people going to get? You will get secondary meristem. When it will again become differentiated, when it will again gain the ability to differentiate, then it will become secondary permanent tissue. Any doubt? Any doubt? So I have given you the examples as well, right? Bache, apical meristem is there, intercalary meristem is there. Here you can take the example of parenchyma, collenchyma, sclerenchyma, your xylem, phloem. Here you can take the example of vascular cambium, cock cambium, and here in secondary permanent tissue, you guys can take the example of secondary cortex. You people can take the example of secondary xylem, secondary phloem. So once I will finish this part, once I'll start the secondary growth, you will understand everything here. So any doubt? Yes, bacho, any doubt? Just a minute. Sure? Are you sure? Pakka? Clear? Crystal clear? That part is important. So now the next is the meristem on the basis of position in the plant body. Okay. So it is on the basis of position in plant body. Clear? Right. So that's what we need to discuss. So the first thing is your apical meristem. What is the meaning of apical? Matlab tips. Okay. It is your apical meristem. So what is the meaning of apical? Right, bache? Right. What is the meaning of apical? It is present at the tips. Yes or no? Yes or no? Right. And second is what? Your intercalary meristem. Okay, your intercalary meristem. Fine. Chaliye, let me explain this. So apical means, see, first of all, you know that it is the example of primary meristem. Yes or no? Hai na? It is the example of primary meristem. So it is the example of primary meristem means it develops during primary growth. Okay, it develops during the primary growth of the plant, right? During initial growth of the plant, this is formed. So it is the example of primary meristem. Secondly, bache, here what we are going to get? We will have root apical meristem. First of all, yes, we'll have shoot apical meristem present at the shoot tip. And another one is root apical meristem present at the root tip. Okay, so shoot apical meristem, root apical meristem. So shoot apical meristem present at shoot tip, present at root tip. Okay, any doubt? Yes, bacho, any doubt? Tell me. Any doubt? This is your apical meristem. So helps in the primary growth, helps in increasing the length of the plant organ. Clear, bache? Helps in increasing the length of the plant organ. Clear? Then intercalary. So what is the meaning of inter? Inter means in between. What is the meaning of inter? Inter means in between. Yes or no? Inter means in between. It is the intercalary meristem. So now what's the point here? Right? What's the point here? Clear, bache? Just have a look. Intercalary meristem. So inter means in between. So it is also the example of primary meristem, no doubt. It is also the example of primary meristem. Fine. But here, what's the scenario? That inter means in between. So it is present in between permanent tissue. Okay. It is present in between permanent tissue. So let me explain how. जैसे ठीक है, simple. जैसे let's say, see here. Like this. Okay. Done? See this. So let's say this yellow part is the permanent. This yellow part is the permanent tissue, and here in between, this part is your meristematic part. This will be your intercalary meristem. So of course you can say it is present in between these permanent tissue. Are you getting it? It is present in between the permanent tissue. Are you getting it? This is the intercalary meristem, right? Bache, jaise suppose karo, you all have seen the sugar cane, right? Right, the sugar cane. So you know that the length of internodes, it varies, isn't it? The length of internodes, it varies. Why? Jaise suppose this is the node, in between these are the internodes. Okay? So here you will find intercalary meristem. Here you people are going to find intercalary meristem. So this meristem is present in between the permanent tissue, and as I said bache, so the difference in the length of these internodes is because of this intercalary meristem. So it is present just above these nodes, and that is why the length is different here. And moreover, जैसे you know about the grasses, right? जैसे grasses के case में sometimes you know grazers, grazing animals, browsers, grazers, right? What are they going to do? They are going to eat the grass, but that grass again grow. How is it possible? It is also possible with the help of intercalary meristem. Are you getting it? It is also possible with the help of intercalary meristem. So any doubt here? Yes. So in grasses, to regenerate the parts removed by grazing herbivores, it is going to help here. Clear, bache? It is going to help here. Done? Sure? Are you sure? Are you sure? So on the basis of position it is, right? On the basis of position it is. So apical meristem we have, intercalary meristem we have, and one more is there, right? Obviously it is as per the position, that is lateral meristem. What is the meaning of lateral? Yes, what is the meaning of lateral? What is the meaning of lateral? I think you know that, isn't it? Lateral means sides, है ना? Lateral means sides. जैसे suppose करो it is a structure. If your tissue is present here, right? These are the sides. जैसे suppose करो these are the lateral side of our body. Dorsal, ventral, lateral. Dorsal, ventral, lateral. Dorsal, ventral, lateral. That is what we have. That is what we have. Yes or no? So lateral meristem is also there, right? When you talk about the meristem on the basis of position, lateral meristem is also there. See, it is present towards the lateral sides. But bacha, you know, there are two things here, right? You know, this part is also given in NCERT, but in NCERT it is written in a very simple line. Okay, see, you are lucky they don't right, they don't ask question from that part, otherwise you will be in the problem. Let me tell you how. Just when you talk about the lateral meristem, obviously it is the meristem which is present, right? It is the meristem which occurs on the, which present on the lateral side of the organ, right? It presents on the lateral side of the organs. Fine, on the lateral sides of the organs, right? Now you know the meaning of lateral sides as well. Now what's the point here, right? What is the point here? Let's see, bache, there are two things when you discuss the lateral meristem. You know, even we have primary lateral meristem also. Do you know that? We have even primary lateral meristem also, and we have secondary lateral meristem. And this secondary lateral meristem is the one, right, that is known as cylindrical meristem. It is the one which is responsible for the secondary growth. Do you know this part? Yes or no? Do you know this part? Students, do you know this part? Yes. See, if I'm using this word, primary lateral meristem, let me tell you, it is again, I'm telling you, it is in the NCERT. It is in the NCERT, but in NCERT it is written in a very simple line, right? Right. So sometimes, you know, you just skip that part. You just cram that part. You don't even understand that. Okay, so let me tell you. So when it is the primary lateral meristem, what is it? When it is the primary lateral meristem, it is lateral, means it is present on the lateral sides. When I'm using the word lateral, means it is present on the lateral sides, but I'm also using the word primary, means it is primary in origin, right? So it is the meristem, primary in origin, which is primary in origin, but present on the lateral sides. Present on the lateral sides. जैसे for an example, intrafascicular cambium. Intrafascicular cambium. Okay, it is intrafascicular cambium. Fine, intrafascicular cambium, and sometimes we even call it as fascicular cambium. let me tell you how will it work. ठीक है, see whenever it is the lateral meristem, always remember it increases girth. Okay, this is the role of lateral meristem. Obvious ना बच्चे देखो, it is very obvious, there is no need to cramp this part. If meristem is present here at the tips, so obviously it will add the cells in this way that there will be the increase in the length of the plant. Yes or no? There will be the increase in the length of the plant. Now if meristem is situated here on the lateral sides, so obviously then again it will add the cell, it is going to do the same job, so obviously it will increase the diameter. It will increase the diameter. So I am using two words. I am saying primary lateral meristem, or I am saying secondary lateral meristem. Right, I am using two words here, primary lateral meristem, secondary lateral meristem. So when it is primary, although it is lateral, it is present towards the lateral side, but it is primary in origin. During primary growth it has formed, right? So I am assuming you know about the vascular bundles, although I will explain it again, but this is what I am assuming, that you already know about the vascular bundles. जैसे we have open vascular bundles. Right, we will discuss about it, don't worry. Jैसे here you will be having phloem, you will be having xylem, and this one is your vascular cambium, or you can simply say that this is your intravascular, sorry, no doubt this is a vascular cambium, or you can simply say that it is intrafascicular cambium. What is it? It is intrafascicular cambium. What is it? It is intrafascicular cambium. So phloem, xylem, in between intrafascicular cambium. Phloem, xylem, in between intrafascicular cambium. Right. So this fascicular cambium has formed during the primary growth of the plant. Okay. So that is why we are saying that it is primary meristem, but it is present towards the lateral side. So that is why I use it as primary lateral meristem. Right. It is the primary lateral meristem. Are you getting it? It is the primary lateral meristem. Fine. So intrafascicular cambium के लिए this is the term that we use, right? Because it has formed during the primary growth. Are you getting it or not? So it is formed from the, it is not the result of dedifferentiation, right? It is not the result of dedifferentiation. It has formed during primary growth. Still there is a doubt, tell me. I will explain this part again. And if you don't know that in your vascular bundles, right, within your vascular bundle you have the cambium. If you are not aware of it, okay, if you are not aware of it, then please let me know, bache, and then please wait for some time. Right, then please wait for some time. We will discuss that too. Okay. Done. So phloem, xylem, and in between cambium. Phloem, xylem, in between cambium. Clear? Clear? So that is why here it is primary lateral meristem. Now when it is secondary lateral meristem, they are secondary in origin. Means they are the result of dedifferentiation of permanent tissue. It is the result of dedifferentiation of permanent tissue. And now here you know the examples very well. Your interfascicular cambium, cock cambium, that is phallogen. But don't be like that, ma'am, this is extra. No, this is not extra. This is not extra. Take it. Tell me. So that is why here we are also using the word cylindrical. Okay, so you are going to see it in the case of dicot stem, in the case of gymnosperms. Fine. Sure. Okay, so interfascicular cambium in dicot stem. Your cock cambium is also there in the dicot stem, and even in the gymnosperms also you will find it. Okay, so secondary growth, because we see secondary growth in the case of gymnosperm stem, You see secondary growth in the case of dicot stem and even in the case of dicot root. So dicot root mein it will be vascular cambium and again the cork cambium, which is the secondary meristem, and here interfascicular cambium cork cambium. हाँ जी बच्चे, डाउट? Done? Sure. So these are the examples. Fine. And I will show it to you from NCERT as well. Done? Okay. So I hope it is clear, bacho. I hope it is clear. So this is the diagram given in your book. And here we can see the apical, the intercalary, and the lateral meristem. Guys, quick, speed up. Yes, speed up. Done. Chaliye. So now this diagram is also given in NCERT. Let's talk about this diagram as well. So bache, here you guys can see they are showing the apical meristem of the root and the shoot here, right? So see, root mein root cap is also there. If you know it, very good. Otherwise, let me draw this part. Just when you talk about the regions of root, it is given in morphology of plants. Okay, so yes, so if you know about the regions of root, so in the regions of root we see the root cap. Okay, we see the root cap, yes or no? And here you will see the zone of meristematic zone. What will you see? What will you see? You will see the meristematic zone, and then here cell elongation will be there. So you will see zone of elongation, yes or no? What you people will see? Zone of elongation, and this one having root here, it is the zone of maturation. So always remember, root here are present in the maturation zone, and here your cells are permanent, they are mature cells, here they are elongating, and here they are actively dividing. So now what am I trying to explain here? Yes, what am I trying to explain here? Here in this diagram, you know that meristem is given. So you see this is the root cap, and here this zone, this part they are showing here. Clear? So see the root cap, the initials of root cap means the one which will form the root cap. Here you guys can see the root apical meristem, and here initials of central cylinder and cortex. Now I will start the sections. I will teach you the sections also. We will take the transverse sections. We will look there in the transverse sections. We will see that how cells are arranged. So then you will understand this part, right? So it is the central cylinder and the cortex. Fine, bache. And here you will see the cortex and the central cylinder. It is nothing made up of tissues and all. I will show you this part. Fine. And here you guys can see, yes, and yes, this protoderm. This protoderm, just remind me, I will explain this protoderm to you. Now, here this is the shoot part. So shoot mein, this is a shoot apical meristem. So when you know, when the shoot grows, this shoot apical meristem also form floral meristem. From this shoot apical meristem, we will also see that floral meristem will also form. Right? Matlab flower basically is a modified shoot. Flower is basically a modified shoot that we are going to see here. And moreover, when it is growing, it is also leaving some part, right? Some part of shoot meristem is left behind. When the shoot meristem is growing, when there is increase in the length, so some part of shoot meristem is also, right, it will left behind. Fine. And that will form these axillary buds. So axillary buds are also the meristematic tissue. Okay, it is also meristematic tissue. Done, bache? And these axillary buds, they are present, you know that, This is the leaf. This is the leaf axis. So it is present at the axil of the leaf, right? So this is the axillary bud. So this bud can even grow and it can even form that branch. Okay, it can even form the branches. Fine. So axillary bud is nothing. It is the left out part of the shoot apical meristem. So it can also develop and it can also form the branches. And here you guys can see the leaf primordia. So is that clear? Yes, bache. Is that clear? Yes or no? Is that clear? Yes or no? Sure? Are you sure? Should we lock it? Arey. Done. So, bache, when we talk about this growth part, na, when we discuss about these growth and all, so we also discuss different theories. Some hystrogen theory, Haber-Lentz theory is also there. There are different theories, but that is not the part of your syllabus. So when we discuss that theories, there are different terminologies. They explain everything, this center, that center, but that is not, again I'm telling you, that is not the part of our syllabus. So I'm not going to pay the attention there, right? But this one word is written here in NCERT, so obviously it's my kind duty to explain you this. So always remember, protoderm is the part that forms epiblemma, or you can say that epidermis. Now if you know epiblemma, then fine, otherwise let me explain you this, right? When you talk about the section of the root hair cells are not covered with the cuticle, right? In the root section you will see root epidermis is not covered with the cuticle. So when that epidermis is not covered with the cuticle, we use the word epiblemma for it. Okay, we use the word epiblemma for it. Are you getting my point? So this protoderm, what is the function of this protoderm? This protoderm is going to form protoderm will develop into the epidermis or epiblemma. Okay, it will develop into the epidermis or epiblemma. So it is as per the Haber-Lentz theory. Okay, it is as per the Haber-Lentz theory. Okay, so it will form epiblemma or epidermis. Okay, so and in the case of, and yes, in the case of, basically in the case of dicots, or in the case of dicots, na, this protoderm also forms, it forms root cap. And remember, not in the case of monocots. Monocots it is little different. So that's what you need to keep in your mind. And why am I telling you? Because this word is given in NCERT, so anytime question can come. That's why. Next. Okay. So meristematic tissue, done and dusted. So let's read it from NCERT, and then we will start the permanent tissue. Okay, so any doubt? Now NCERT reading time, NCERT reading time. Okay, so if there is any doubt, please tell me, bachcho. Right? If there is any doubt, please tell me. Sure? Sure? Okay, so see, as per NCERT, let's discuss it as per NCERT. So growth in plant is largely restricted to specialized region of active cell division, which are known as meristem. You know that plants, they grow throughout, right? In the case of plants, na bache, when we use this term growth, I will teach you this part in the case of plant physiology. Plant may growth is open type. Do you know that? Plant may growth is open type. It is given in plant growth and development portion. What type of growth is there, Raghav? It is the open type. What is the meaning of open type of growth? The open type of growth means the cells will be added by the meristem, right? Further cells will be added by the meristem. Are you getting it? Right? That meristems are going to add the cells, and so it is the open form of growth. It can be determinate, it can be indeterminate, like it is continuous throughout the life. Why? Because of the presence of this meristem. Because meristem is adding the new cells. That is why we use the word open type of growth in the case of plant cells. Okay. Okay. Take it. So plants have different kind of meristem. I told you already. So if it is present at the tips of root and shoot, basically if it is present at the tips, root apical meristem or shoot apical meristem. Apical means tip. Okay? So root apical meristem, in short form, we used to call it as a ram. So it occupies the tip of root, and SAM, shoot apical meristem, it occupies the distant most region of the stem axis. Fine. So during the formation of leaves and elongation of stem, some cells they left behind, and they are the one which will form the axillary bud. So can I say that axillary bud Huh. So can I say that even the axillary bud is meristematic? Of course it is. Right? Of course it is. So such buds are present in the axials of the leaves and are capable of forming a branch or a flower. Right? So every time when you look at a leaf, when will you say that it is a leaf? When the axillary bud is present here. Okay. Okay, that is how we even differentiate simple leaf from the permanent leaf. Done, bache? Done, bache? Sure. So meristem which occurs between mature tissue is known as intercalary meristem. The meristem which occurs in between, right, which occurs in between mature tissue is known as intercalary meristem. So they occur in grasses and regenerated parts removed by the grazing herbivores, but basically they help in that. Okay, so both of them are primary meristem because they appear in the primary growth of the body. So now see, meristem that occurs in the mature regions of root and shoot, right? What are they? What are they? Right? See, they are helping in the growth. They are helping in increasing the girth, right? See, that produce woody axis and appear later than primary meristem, that is secondary or lateral. Any doubt here? Any doubt here? Some students in the comment section, they were saying ki ma'am, we want the NCERT line to line. So in every session of mine, I always explain the topics from the NCERT as well. Okay, so is that clear? So these are the cylindrical meristem. So which meristem is cylindrical? Which forms later than primary meristem? Simple as that. Okay, so fascicular vascular cambium, interfascicular cambium and cock cambium are examples of lateral meristem. Let me tell you this point here. Fascicular cambium means intra mane within. So intrafascicular cambium and fascicular cambium is same. Right? Don't make the mistake here. Intrafascicular cambium and the fascicular cambium is same. Please don't make the mistake here. That's very important, Suhashini. Okay, that's very, very, very important. So these are responsible for producing the secondary tissue. So I told you already that this fascicular cambium, it is primary lateral meristem. Rest two examples are secondary lateral meristem. Clear, bache? Rest two examples are secondary lateral meristem. So they are responsible for producing the secondary tissue. So see, following division of cells in both primary as well as secondary, newly formed cell will become specialized. They will lose their ability to divide. They are the permanent or mature cells. Clear, bache? Any doubt? Any doubt? So now what is going to happen? During the formation of primary plant body, specific regions of the apical meristem, they produce dermal tissue, ground tissue and vascular tissue. That is what we need to discuss now. So if there is any doubt, please let me know, because after it we will start with the permanent tissue. Okay? Right? So today's session will be, obviously, session will be till 10. It can go till 10. Okay? So I will not rush. In between, I will give you the breaks so that we can go in one flow. Okay, I don't want, bache, I really don't want ki, theek hai, I'm just teaching and you have just, started the video and you are not getting anything. Literally, I don't want that. So in between, I will give you the break. So we will get 3 breaks, 1 break, 5 to 6 minutes ka tea break. Again, another 5 to 6 minutes ka break for the dinner. And if required, one another break. Otherwise, 2 breaks will be given to you. And again, I'm telling you this thing, bache, that please join this batch, right? Join this batch started by your HSP sir. Bache, HSP sir is actually the god of chemistry. He's a very, very, very good teacher. Okay? Literally, he's a very good teacher. And do you know, even I'm thinking that I should give the NEET examination this year. So, bio to I can manage. So, I'm thinking that I should start. I just want to give the biology exam and I will see how many marks I'm getting from that biology portion. But honestly speaking, I'm thinking to prepare two chapters of physics and chemistry as well. है ना? Two chapters of physics and chemistry as well. What will you suggest to me? Just suppose karo, even I am thinking to start the NEET preparation. Okay, I am thinking to start the NEET preparation. So you tell me that at least tell me not 2 chapters, 2 units. 1 unit of chemistry, 1 unit of physics. That's all. This is what I can prepare. What will you suggest? Arey, we can prepare together, but whenever I'll get the free time, then only I'll study that physics and chemistry, no? So as of now, just tell me As of now, just tell me 1 unit of physics, 1 unit of chemistry. ठीक है? 1 unit of physics, 1 unit of chemistry. Organic chemistry, I don't like organic. My personal, I like physical chemistry, I like inorganic chemistry. I don't like organic. I don't know why. ठीक है, मतलब I will take classes from HSP sir and Wasim sir. I will take classes from HSP sir and Wasim sir. You tell me 1 unit. I'm not asking for one that organic, inorganic and physical. नहीं भाई, सिर्फ एक unit, only 1 unit. Thermodynamics, okay? मतलब I biomolecules of chemistry or that I know. Electrostatics and physics. ठीक है. I'll think of it, huh? If I'll start preparing physics or chemistry, I'll tell you in my Telegram group. You guys can join my personal Telegram as well, Love Biology by Ambika. Okay, so here you guys can see our Guruji has started the batch Chemistry Mind Maps by HSP sir. So just click the link, and what you have to do, click on the link, add your number, add your email ID. You will get this batch at a price of only 1999. Right? So join it ASAP. Okay, join it ASAP. Do you want something like this in biology as well? Huh? Tell me, do you want something like this in biology as well? Ma'am, imagine Wasim sir and Shreya sir reaction when you tell them you are only going to prepare one unit for NEET exam. They are be like, "Hamara naak katwaegi. You're useless." They'll be like this. You're useless. You're only preparing 1 unit. You're our friend. You have to prepare 50% of the syllabus at least. I'm not going to tell them. Literally, I'm not going to tell them. ठीक है? So if I need the help, then I'll call them कि ठीक है मुझे बताओ. Otherwise, I will check their YouTube sessions. है ना? I will check their YouTube sessions. So electrostatics and what you just said, biomolecules तो हो जाएगा. Electrostatics from thermodynamics. ठीक है? Let's see. When I was in class 11, then I was not able to understand thermodynamics. I was not. Now I will try. I will try my best. ठीक है? चलो. So I am giving you a break, and this time है ना, I will start it on time for 5 minutes. Just for 5 minutes. So it is a kind of washroom break. ठीक है? It's a kind of washroom break. Fine. So till then I will get my what? Cup of tea, of course. That's what I want. And if I will prepare thermodynamics, I will ask questions from you. I was thinking that I should prepare something from inorganic chemistry. Science of exceptions. Ha, Shiva Kumar, I'm thinking that I should give it a try. Like biology I can manage, but if I have to sit there for 3 hours, so I should give some time to physics and chemistry too. Chalo, theek hai. We'll meet you after 5 minutes. Hi Maya, है ना Maya? Imagine a teacher is saying कि यार I just need one unit of physics and chemistry to clear the NEET, but actually I cannot. But this is what I am going to prepare. So if this year, if I'll give the exam, then I will go for 1 unit of physics and 1 unit of chemistry as well. ठीक है? This is what I'll do. ठीक है? Okay. चलो. So can we start the next part? Imagine I'm giving the NEET exam and you are sitting next to me and you're like, bam, bam, bam. Please tell me what is the answer of this question of biology. And then we both will be disqualified. Examiner will throw us out. Nahi yaar, Maya, some students they don't even prepare a single unit yaar. I'm at least preparing single unit. Chalo. So now let's start the next part, that is the permanent tissue. Okay? That's what we need to start. What we need to start? We need to start the permanent tissue. So I hope you are enjoying today's session flow. Hannah, today we are talking in between, we are discussing one topic, and you are getting a break as well, right? Even a short break, but still you are getting a break as well. That's good now. That's good now. Chalo. So now when it comes to the permanent tissue, here cells, they are not dividing. Hannah, they are not dividing. They lost ability to divide, isn't it? They lost ability to divide, isn't it? Permanent tissue. And second part here is, take a cells, they have lost the ability to divide. This is the meaning of permanent tissue. So in the permanent, and they are performing a particular function now. Henna. So now here we have two things. One is simple permanent tissue, bache. One is simple permanent tissue, and the second one is complex permanent tissue. One is simple permanent tissue and the second one is complex permanent tissue. Now when it is the simple, it is made up of one type of cell, right? It is made up of one type of cell. Like you can say that same type of cells are there. Done. And here it is made up of different types of cells. That is why it is complex. It is made up of different type of cells. That is why it is what? It is complex. Clear, bache? Clear? So here in simple permanent tissue, we have three examples and I hope you know about it. That parenchyma, collenchyma, that's collenchyma. And third one is sclerenchyma. Okay, sclerenchyma. And here you will see xylem and phloem. Xylem and phloem. Clear, bache? Xylem and phloem. So what do we have here? Just have a look. Right, permanent tissue cells, they have lost the ability to divide. They are performing a particular function. What are they doing? They are performing a particular function, right? So in permanent tissue, you will see living cells as well, or the dead cells as well. It is not mandatory that all the cells are going to be living here. They can be dead as well. But in meristematic cells, they are dividing. Obviously, they all are the living cells, isn't it? So here, simple permanent tissue, complex permanent tissue. Simple permanent tissue, complex permanent tissue. Simple permanent tissue mein, it is made up of one type of cell. Complex permanent tissue mein, it is made up of different types of cell. Parenchyma, collenchyma, sclerenchyma, xylem, phloem. Parenchyma, collenchyma, sclerenchyma, xylem, phloem. Guys, doubt? Yes, any doubt? Students, be quick. And even you have to make the notes, right? Do not just wait for my PDF. Even you have to make the notes. Clear, bache? Clear, bache? Guys, quick, quick, quick. Sure. So next is our simple permanent tissue, where we are going to discuss the parenchyma first. Take it. So in simple permanent tissue, having one kind of cells only. So what are we going to start with? We are going to start with the parenchyma. We are going to start with the parenchyma. Okay, we are going to start with the parenchyma. So parenchyma, it is the first permanent, or you can say that first differentiated tissue. It is the first permanent or the first differentiated tissue, bache, right? So majority of organs of a plant will contain parenchyma, right? So parenchyma is present in majority, right? Parenchyma is present in majority. So it is the first permanent or the differentiated tissue, bache. Clear, bache? Clear, bache? So you will see that pulp of fruit contains parenchyma. So majority में in a plant we will see parenchyma. And bache, do you know, first of all, let me talk about its cell spaces, its structure. Bache, you will see here in this tissue, whenever you will see the cells, right, they will be, no doubt in diagram they are not looking like, but you will see that cells can have different shapes. They can be oval, they can be polygonal, like any shape can be there. But one thing that you need to focus here is that they will be isodiametric, right? That all the cells here in this tissue, they are isodiametric. Iso means same, diametric. Isodiameter means same diameter is there. Okay, isodiametric means same diameter will be there. Are you getting it? Simple permanent tissue having one kind of cells only. So we are starting with what? We are starting with the parenchyma, bache. So it is the first permanent and differentiated tissue. So the pulp of fruit will contain parenchyma. And here you guys can see, here you people can see the cells are having different shape. They can be oval, they can be polygonal, etc. And here you will see that they are going to be isodiametric, right? They are going to be what? They are going to be isodiametric. Any doubt, bache? What are they? They are going to be, yes, bache, they are going to be isodiametric. And now if I talk about, just have a look of that diagram from NCERT, right? So here you will see that these tissues, it is what, right? Wait, it's a compact tissue. It's a compact tissue, right? It means they have no intercellular spaces, or if spaces are there, or you can say that very less intercellular spaces are there. Are you getting my point? Very less intercellular spaces are present. That's what you need to keep in your mind. Okay, done. And when you talk about the wall of the cells here, cell wall here is what? Cell wall is thin, right? It is thin, made up of pectocellulose. It is thin, made up of pectocellulose, like pectin and cellulose is there. Are you getting my point? So thin cell walls, right? So in the case of parenchyma, the cell wall is going to be thin. So what is the meaning of this particular statement? That obviously they are making the soft part of the plant, right? It is not meant for the mechanical support. Okay, it is basically making the soft part of the plant, and you will find them in the cortex, in the pericycle. You will see the cells majorly. What are they? They are parenchymatous cells. Are you getting my point, bache? So they are isodiametric. They can be oval. They can be polygonal, right? So thin cellulosic cell wall is there, bache, right? Thin cellulosic cell wall is there. So I hope this part is clear to you, right? So it is not at all difficult. Fine, they are not at all difficult. So thin cellulose, thin cell wall is there. It is made up of pectocellulose, or you can simply write the cellulose. So it is there in the softer part of the, right? So you will see it in the softer parts of plant. Softer parts of, you can say that stem, leaves, fruits will contain your parenchyma. Okay? So it's a living tissue. Fine, it's a living tissue. Any doubt? Yes, bache. Any doubt? Yes or no? Do you have any doubt? So bache, they have the parenchyma cells, they have different function, right? Like parenchyma tissue, It performs different function, right? So it is also, so mainly it is for the storage of food. The main function, if I have to talk about the main function here, so it is mainly the storage of the food. So as I said, parenchyma, it helps in the storage of food, mainly in the storage of food. And do you know what? That parenchyma cells, right? This tissue can be photosynthetic also. This can be photosynthetic also. Right, and sometimes even some waste products are stored here in parenchyma, right? Some waste products are also stored here in parenchyma. So when it comes to the parenchyma, it is having different functions, like photosynthesis is also the function, storage is also the function, right bache, right bache. So sometimes it is also, right, sometimes it is also helping in the, you can say that, in the secretions as well. Yes, parenchymal cells, some cells can be secretory as well. They are even playing role in the secretion as well. So I'll give you 1 or 2 examples here. Yes, I will give you 1 or 2 examples here. Jaise parenchyma. Do you know about parenchyma? Do you know about parenchyma? Yes, these are the modified parenchyma tissues. Right, these are the modified parenchyma tissues, right? So it is basically, parenchyma is basically the network of, it is the network of parenchyma cells having large air spaces. Fine, they are having large air spaces, like air spaces are present, cavities are present. So obviously it is meant for the buoyancy. Okay, it is meant for the buoyancy. So parenchyma, what is it? It is the network of parenchyma cell having large air spaces or large cavities are there, right? Large air spaces or large air cavities are there. So bache, you will find it in the case of aquatic plants. So its main role is to provide the buoyancy. Clear? Right? It will provide the buoyancy. Are you getting it? It is going to do what? It will provide the buoyancy. Same way, Sclerenchyma. Do you know about Sclerenchyma? Sclerenchyma. It's not Collenchyma, it's Sclerenchyma. Now here, your parenchyma is performing photosynthesis. Right? Your parenchyma is performing photosynthesis. Here, in Sclerenchyma, you will see that cells of parenchyma, right? The cells here, they are rich in chloroplast. So if they are rich in chloroplast, means they will do what? They are going to perform the photosynthesis. Am I right? So cells are rich in chloroplast. Cells are rich in chloroplast here. Clear, bache? So can I say that in the case of leaves, जैसे when you talk about the leaves के case में we have the mesophyll. When you talk about the internal structure of the leaves, we have the mesophyll. It is basically the ground substance of a leaf. So that mesophyll cells are nothing. These are these. They are enriched with the chloroplast. They are basically, the parenchyma is the Sclerenchyma here. Right? So Collenchyma is different. It is Sclerenchyma. Okay? It is Sclerenchyma. Okay? What is it? It is Sclerenchyma. Clear, bache? Are you sure? Yes? Are you sure? Yes or no? So sometimes, you know, one more example is there, that is idioblasts. Do you know the meaning of idioblasts? So they are also some parenchyma cells, na? Some parenchyma cells. Fine. So here what's the point? That some parenchyma cells, they get modified and store oils, tannins, right? Like this, ergastric substances rather. Right? So sometimes some parenchyma cells, they even store, right, bache? They even store these ergastric substances, means these substances are inert, right? Like oils are there, tannins are there, right? Oils are there, tannins are there. It can be gums, raisins, anything can be there. So when the parenchyma cells, they start storing oils, tannins, even the raisins, even the gums, they all substances, they are what? These substances are metabolically inert, Metabolically inert means they are not taking part in that chemical reactions. They are not reactive basically. They are not reactive basically. Such cells are idioblasts. Clear, bache? Such cells are what? Such cells are idioblasts. Is that clear? Is that clear? Now, bache, do you know about stomata? Do you know about stomata? Anyone here in the class? Do you know about the stomata? है ना? Stomata, you know na? Pore-like structure from where the gaseous exchange will be there and the loss of water in the form of vapors, that is known as transpiration. So if you know about the stomata, then I'm pretty sure you even know about the guard cells. You even know about the guard cells. So these guard cells are also having the chloroplast. And let me tell you, these guard cells are also the parenchymatous cells. So see, parenchyma is modified in different ways. है ना? Parenchyma is modified in different ways. So these guard cells are also the parenchyma cells. So this is about the parenchyma. These features are important. Isodiametric, the shape can be oval, polygonal, it can be spherical as well. Okay, and cell wall is going to be thin here. Clear? So that's how it works. So up to this part all clear? Yes, mera bacha. Up to this part all clear. So parenchyma is making the soft tissue. So here you guys can see, right? So this tissue is the compact tissue. Fine, it is compact. If there will be the intercellular spaces, they are less, right? They are less. Fine. Is it clear? Is it clear? So just have a look, bache. Let's revise it from NCERT. So a simple tissue is made up of only one type of cells, right? So parenchyma forms the major component within organs. I told you already. So cells here are isodiametric, very important to remember. They may be spherical, oval, round and polygonal, or they can be elongated in shape, right? They can be elongated in shape. Just in mesophyll, your palisade parenchyma, the cells are elongated there. Okay? Walls are thin, made up of cellulose. Told you already. They are closely packed, or they can have small intercellular spaces. Done, bache? So parenchyma can perform different jobs. I told you already. Next that we need to discuss is your collenchyma. This is the diagram from NCERT. Okay? Next is what? Collenchyma. So it provides the mechanical strength. Okay, so it is meant for the support. So if this tissue is meant for the support, so obviously the walls here are going to be thick in comparison to the parenchyma. There is no need to cram. You can understand it in this way, and that's very simple, as I said. That's very simple, as I said. So what is going to happen here? What is going to happen here, bacho? Here you will see that cell walls are going to be thick. Here in the sides you will find some thickenings, bache, right? You will find some thickenings here. Diagrams are important. Please focus here. Okay? So when it comes to the collenchyma, so they are meant for the protection, and you will see the cell walls are thickened, and here, just a minute. By deposition of, here it is not just the cellulose, hemicellulose and pectin is also there. So along with cellulose, we will even see hemicellulose. We are even going to see the pectin. Okay. And let me tell you, bache, जैसे now we will start the sections. We will talk about the sections. ठीक है? जैसे suppose करो, this is a stem. I am taking its cross section. So when I will take its cross section, I will study the different types of tissues there. So you will see that outermost layer is going to be epidermis. Let me show you here, right? That, see, let's say this is the TS. This is the TS of anything. Okay, it is the TS of anything. Let's say it is the TS of dicot stem. So obviously outermost layer that we are going to consider is going to be the epidermis. है ना? These are the basics. Yes or no? These are the basics. Outermost layer will be what? Outermost layer is going to be epidermis. So below this epidermis, right? Just below this epidermis, what will you get? You will get hypodermis. Hypo means below, right? Hypo means below. So you will see that these collenchymatous cells, that's important, huh? Collenchyma is making hypodermis of, dicot stem. Do you know that? It is making hypodermis of dicot stem. Now, what is the meaning here? That, see, note down, this is important, bache, kindly highlight this part, that collenchyma is absent, right? It is absent in roots. Like in roots, you are not going to see collenchyma cells, and in monocots. That's important. That's why, bache, I am highlighting it. That's important. Collenchyma is absent in roots, and it is even absent in your monocots. Clear? So in the case of dicot, especially in the case of dicot stem, you will see the hypodermis contains collenchyma cells. You will see the hypodermis contain collenchyma cells. Is that clear? Yes, bache. Is that clear? And the cells there are what? They are thickened. Clear, bache? Clear, bache? Yes or no? So it is absent in roots. It is absent in monocots. It is absent in roots. It is absent in monocots. So can I say that it is present in the dicot stems? Yes. Can I say that it is present in the dicot stems? Yes or no? So cells, they have the thickenings on the cell wall, and yes, they are absent in the roots and monocots. Clear, bache? Clear, bache? Yes or no? Quick. Okay. Sure? Done. So here you guys can see, and yes, the collenchyma, the cells are living. It is also not a dead tissue. Lignin is not there. It is living. So you will see that thickenings here are because of cellulose, hemicellulose, and pectin. Okay, bache. And yes, it is because of cellulose, hemicellulose, and pectin. And I told you already where is it present or not. Okay. And you know collenchyma, if it is having chloroplast, then obviously it will be photosynthetic. Then it will be photosynthetic, if right. Done, bache. So its main function is to provide the support, is to provide the flexibility as well. Right? Is to provide the support, is to provide the flexibility as well. Clear, bache? Yes or no? Tell me. Tell me. So major function is of support, flexibility, elasticity as well. Okay? Done? Sure? Sure, bache? So it is living. Right? And collenchyma, maybe you will not see no intercellular spaces. No intercellular spaces are there. So when it comes to the functions, so functions may, as I said, support, flexibility to the soft parts. And do you know, jaise bache, you know that they are present, yes, whenever there is the, whenever there is a strong wind, actually this collenchyma is going to protect that stem. Do you know that? It is the collenchyma which is going to protect that stem. Fine. It is the collenchyma which is going to protect that stem. This is what you need to keep in your mind. Done, bache? So let's revise it from NCERT, and yes, do not forget this particular point that they are absent in roots and in monocots. So see, you can see they occur in layers below the epidermis. So below the epidermis means hypodermis will be there. Yes or no? And here in NCERT they have just mentioned dicot stem. So I told you they have just mentioned dicot. So only in dicot stem you will find it. Ultimately in roots and monocots they are absent. That's what you need to keep in your mind. Okay, it is absent. Done, bache? So it can either present as a homogeneous layer or in patches. Homogeneous layer means uniformly distributed. This is, suppose karo here you are having the collenchyma, a proper layer is there, right? A proper layer is there. Or as I said, it can be in the form of patches. This is, suppose karo one patch is present here. We will see in bundle sheath cells. Another patch is present here. Like that, layer is not continuous. It is present in patches. Okay, Arthi? It is present in patches. So it consists of cells which are much thickened at the corners. Corners mein thickening is more. Why? Because cellulose, hemicellulose and pectin, they are deposited. So shape can again vary here. It can be oval, spherical or polygonal, and it contains chloroplast. So often contain chloroplast. So if it is containing chloroplast, then obviously it will perform photosynthesis. Clear bache? So they will assimilate food if chloroplast is there. So it will provide mechanical strength to the growing part such as young stem and petiole. And as I said, it will also protect your stem from the bending as well whenever there is a heavy wind. Okay, so doubt? Hanji, doubt? Quick. Done? Sure? So that is what you need to remember. Okay? Okay. So next is the Sclerenchyma, and when it is the Sclerenchyma, obviously it is also going to provide the mechanical strength. And one thing that you need to remember here is that here it is what? This Sclerenchyma is dead. Okay? So here the cell walls are going to be lignified. So whenever we use the word lignified cell walls, right? So always remember there is no protoplast and the cell is dead. Is that clear? And the cell is dead. Clear, bache? So just have a look. Sclerids, right? So Sclerenchyma. So what will be discussed here? Sclerids and fibers. Sclerenchyma mein sclerid and fibers. Clear? Yes or no? Sclerids and fibers. So Sclerenchyma, they have thick secondary walls. They are dead. They have pits, right? So they have two forms. Take it. Sclerenchyma. It is, Sclerenchyma is having thick secondary walls which are lignified. Lignified means lignin is present there. Okay, and this tissue, here the cells are what? Dead. Dead tissue it is. And why is it dead? Why is it dead? Why? Because of the deposition of the lignin, of course. Okay, so here you will see in Sclerenchyma, we will see some simple pits are also present. Okay, simple pits are also there. Fine. And obviously it will also provide the mechanical support. What will it do? It will also provide the mechanical support. Clear, bache? And here you will see two forms in Sclerenchyma. This one is the fiber and this one is the sclerid. Okay, this one elongated, tapering is the fiber. This one circular, having pits canal, this is the sclerid. So the diagram can be asked in the paper. Okay? Okay? Clear, bache? The diagram can be asked in the paper. First of all, note down. So on the basis of variation in form, we have two types of Sclerenchyma, fibers and the sclerids. Right? Fibers and the sclerid. Okay? Okay. So pits are overall present. Okay, bache? Pits are overall present. Sure. So now let's talk about the fibers first. So when you talk about the fiber, okay, so fiber is what? You can just look at the diagram. The cells are elongated, they are tapering. Yes or no? Elongated, they are tapering. Can you see it? In the case of fibers, the cells are what? They are elongated, they are tapering. Okay, they are tapering or they are pointed. What are they? They are tapering or they are pointed. So, bache, see, the cells, they are, the one thing is for sure, that cells, they are thick walled, they are elongated and they are going to be pointed. Fine. They are going to be pointed. Fine, bache. These are your, yes, these are your fibers. Thick walled, elongated, pointed. Thick walled, elongated, pointed. Right? Repeat it, bache. Thick walled, elongated, pointed. Clear, bache? Clear, bache? So here you are going to see these pits, these spaces here. right? Can you see these pits here? Let me change the color. Right, can you see these pits here? These are the simple pits basically. What are they? They are the simple pits basically. Okay, what are they? They are the simple pits basically. So elongated, pointed, thick walled structure it is. Obviously it is also providing the mechanical support, bache, right? It is also providing the mechanical support. And where are you going to find these fibers? Let me tell you, right? Fibers you guys are going to see in the case of hypodermis of monocot stem. Do you know that? Hypodermis of monocot stem will contain is having the sclerenchyma. These are the important points, bacho, right? And one should not leave such points, right? So hypodermis of monocot stem will have sclerenchyma, right? Hypodermis of monocot stem will have sclerenchyma, and it is basically the fiber. Clear, bache? It is basically the fiber. Okay. Okay. And do you know we are also going to talk about the vascular bundles, matlab bundles of xylem and phloem. You will see that vascular bundles are also covered, right? They are having a bundle sheath in the surrounding. Just these fibers, yes, these fibers, they are also making that bundle sheath of vascular bundles. That I will explain you later. Okay, they will even form the bundle sheath of vascular bundles. Is that clear? It is also forming the bundle sheath of the, yes, bundle sheath of the vascular bundle. Clear, bache? Yes or no? Yes or no? Done. Sure. So we have the phloem fibers also, we have the xylem fibers also. Means in the xylem also fibers are present, in the phloem also fibers are present. So xylary fibers are also there, phloem fibers are also there. Let me tell you. So we have the xylary fibers also, like in xylem we have the fibers also, and even in the case of phloem also you will see the fibers. Like xylem and phloem, they are complex tissues, they are made up of group of cells, so they also contain the fibers. That's what we are saying. Okay, that's what we are saying. Is that clear? Is that clear? Yes or no? Is that clear? Just now I will give you the example of phloem fibers. Phloem fibers mein you have the example of vast fibers. This vast fibers, you know, they are obtained from the phloem. This is important, bacho. They are obtained from phloem. Jaise, do you know about cannabis, hemp plant? Cannabis से we are also getting the fibers, right? जैसे cannabis हो गया, hemp plant. Other than that, jute is also there, right? Jute is also there. You know that this hemp, this jute, we use it, isn't it? This cannabis, this jute, we use it for different purposes. Like they are having the commercial purpose. So they are the example of vast fibers. So what are the vast fibers? They are obtained from the phloem. So basically they are the phloem fibers. Clear? They are the phloem fibers. Are you getting it? They are the phloem fibers. So these are the fibers which are present in phloem. So cannabis is there, jute is there. Clear? Cannabis is there, jute is there. Okay. Sure, bache? Right? Clear? Bache, let them spam. Why are you wasting your time? Why are you wasting your time? They are the spammers. They don't have anything else to do. So this is what they used to do. Why are you wasting your time? You just focus here, na? Okay? So that's what you need to remember. Now next is what? Next is the sclerids. Next is what? Next is the sclerids that we have. So just look at this diagram. So here you guys can see they are having the pit canals. See, right? There is a lumen within. Look at the lumen here. So sclerids are highly thickened, broader with narrow cavity. Isn't it? Isn't it? They are highly thickened, broader, and they are with narrow cavities, and of course they are also dead, lignified, hai na? Dead, cell walls are dead and it is lignified as well. Cell walls are dead, it is lignified as well. Clear, bache? Clear, bache? So they can be branched, they can occur singly, they can occur in the groups as well. Fine. So even their function is to do what? Support. है ना? So you will see, you will find them commonly in nuts, है ना? In fruit nuts, right? This, are you getting it? ठीक है? So commonly Where will you find it? Commonly in the fruit walls of the nuts, right? In the pulp of the fruits. Done. So there basically their examples are important. So commonly in the fruit walls of nuts, in the leguminous plants also, just your leguminous plants also, they also get these sclerids. In the sapota, right, guava, they also contain these sclerids. So that is what you need to remember. Clear, bache? Clear. Okay, so sclerids may, you know that pit canals are there basically. So now let's revise it from the NCERT. Fine, let's revise it from the NCERT. So Sclerenchyma, having long narrow cells with thick and lignified cell walls, having a few or numerous pits. So when we talk about the sclerids, always remember in the case of sclerids, yes, always remember in the case of sclerids, what will you get? What will you get? You will see the pit canals there. Clear? You will see the pit canals there. So they are usually dead without protoplast. So obviously, bache, when they attain the maturity, at that time they are dead. Initially, so these cells are living, and when they will become dead, right? They are without protoplast. Clear, bache? So on the basis of variation in form, structure, origin and development, Sclerenchyma may be either fibers or sclerid. So fibers are thick walled, elongated, pointed. Thick walled, elongated, pointed, and generally they occur in the groups, right? So sclerids, what are they? They are spherical, oval, cylindrical, highly thickened dead cells with narrow cavity, narrow lumen. So they are in the fruit wall of nuts. Pulp of fruits like guava, pear, sapota contains it. Seed coats of legumes and the leaves of the tree. This part is important, right, bache? These examples are important. Any doubt? Okay? Okay? Clear? Sure? So next is the xylem and phloem, and xylem and phloem is what? They are the complex tissues. है ना? Xylem and phloem, so what are they? They are the complex tissue. So that's it. This is about the simple tissue. Bacho, please do not waste your time. So tell me, up to this part all clear? Sure? Take up to this part all clear? Yes? Okay? Done, bache, any doubt? Okay. Chali. So the next is the complex tissue, xylem, phloem. Xylem, phloem, and you know that they are very important, xylem and phloem, isn't it? जैसे देखो, in the case of plants, yes. Now, in the case of plants, you know that plants, they do not have any heart. They do not have a proper circulatory system. Plants may, they do not have any heart. They do not have a proper circulatory system. So, in the case of plants, how that transportation is possible? In the case of plants, bache, when it comes to the long distance transport, right? When what is it? When it is the long distance transport, right? For long distance transport, we need xylem as well as phloem, right? So xylem and phloem, they are together going to form the vascular bundle in the case of plants. What are they going to form? They are going to form the vascular bundle in the case of plants. And let me tell you, bache, for long distance transport, yes, we need xylem and phloem. And when xylem and phloem, right? For xylem, I will use X, and for phloem, I will use P. So when xylem and phloem, right, they are involved in transportation, the word is translocation. Okay, when they are involved in transportation, okay, the word is translocation. What is it? It is translocation. Are you getting it? It is translocation. Clear, bache? And now the xylem, it helps in the transport of water and minerals. Right? So water and minerals. In water, that minerals are mixed. So it is a sap basically. So xylem helps in the transport of water and mineral, and xylem movement is bache unidirectional. Right? So from roots, the absorption of water and minerals will be there, and then from root it will pass to the stem, it will pass to the leaf. So the movement is unidirectional. Okay? In the case of xylem, the movement is unidirectional. And when you talk about the phloem, so it is meant for the transportation of food. Right? It is meant for the transport of food. Right? So phloem mein, the movement is going to be bidirectional. Why? Because in leaves there will be the synthesis. Okay, so why? Because in the leaves, there will be the synthesis. There will be the synthesis of food, and then from leaves it will be translocated to the different parts. Okay, so the food will pass to the other branches also. It will pass to the flowers. It will pass to that stem also. Take it to that root also. So here in the phloem, the movement of substance is bidirectional, and here it is going to be unidirectional. So xylem and phloem, what are they? They are the complex permanent tissue because they are made up of different type of cells. Right? More than one kind of cells are there. More than one kind of cells are there. Right? More than one kind of cells are there. Done, bache? Clear? Clear? So when the xylem and phloem is involved in the transportation, then the word is, yes, it is the vascular bundle, and the word is translocation then. The word is translocation then. Clear, bache? Clear, bache? So now when you talk about xylem, so xylem is made up of, yes, it is made up of tracheids, then vessels, then xylem fibers are there. See, xylem fibers are there, and then comes the xylem parenchyma. Then comes what? Then comes the xylem parenchyma. Yes or no? And when it is the phloem mein what do we have? Phloem mein, yes, sieve tube, companion cells, phloem parenchyma, phloem fibers. So these three here, they are dead. Dead means lignified, of course, without protoplast. And xylem parenchyma is the living here. Okay, it is the living here. Now when you look at this part, right, when you look at the phloem part, just say here you have the sieve tubes, companion cell, phloem parenchyma. These three are living, right? These three are living tissue. This one is dead here. Okay, this is the first question, right? First question from this topic. Fine, this is what they can ask you. So when you are talking about the xylem made up of tracheid, vessel, fibers and parenchyma, so tracheid, vessel, fibers, they are dead, parenchyma is living. In the case of phloem, you have sieve tubes, companion cell, phloem parenchyma, phloem fibers. So sieve tube, companion cell, phloem parenchyma, they are living, phloem fiber, it is dead. Is it clear? Phloem fiber, it is dead. Is it clear? ठीक है? Okay. So tracheids, vessels, fibers, parenchyma, sieve tube, companion cells, parenchyma or the fibers. ठीक है? So clear? चलो, now let's start the xylem. Xylem. चलो. So बच्चे, yes, one more thing. देखो, tracheids and vessels. देखो, listen to me, huh? These tracheids and vessels, they are mainly helping in the water transportation. Do you know that? These tracheids and vessels, they are mainly helping in the water transportation. They are the tertiary elements. Both of them are considered as the tertiary elements because they are the one that transport water. Okay, they are the one that transports water. Okay, they are the one that transports water. The tracheids and the vessels, tertiary elements, they transport the water. Then comes the xylem fiber and parenchyma. They are mainly for the parenchyma. You know the role of parenchyma, and the fibers is there to provide the strength. But mainly for the water transportation, these tracheids and vessels are going to help, and that is why we use the word tertiary element for it, right? That is why we use the word tertiary element for it. But when you talk about the phloem, sieve tube, companion cell, phloem, there also we need to discuss few points. But first of all, this is what you need to keep in your mind. Clear, bache? This is what you need to keep in your mind. So now what are we going to start? We are going to start with the tracheids, which are elongated or tube-like, thick and lignified cells. Right? What are they? They are thick, tube-like, lignified cells. Okay? So doubt? Yes. Any doubt? Just a minute, guys. Allow me a minute, please. Okay. So water and mineral will be conducted by your xylem. Done. Chalo. So tracheids and vessels are there, and these are the tertiary elements. Now, tell me one thing. Let's relate it with the plant physiology a bit. I'm saying that in the xylem, tracheids and vessels are going to transport the water. They are helping in the transportation of the water, and they are dead. Do you think that there is any significance in this point? That why are they dead? Like, why? What's the reason? Why are they dead? Tell me. Do you find it significant if they are dead? Dead means without protoplast. There is no protoplast there in the xylem. And the tracheids and vessels and the fibers, of course, xylem parenchyma is again living. Give it a try. Give it a try. Whatever reason is there in your mind, you can share it with me. Just I told you that here in the xylem, we have the tracheids, we have the vessels. So I am saying that, they are dead. So is there any significance? Is there any significance? Yes, is there any significance? Give it a try. Just think of it. But of course, देखो, root hairs will absorb water and water will go up. So this is basically the capillary action. Here the diameter is going to be very less. These are basically the thin tube. This is basically the capillary action. Transpiration pull will be there and the water will pass like this. Okay, it is a kind of advantage here, right? That is why this part is basically dead. The tracheid and vessel, this tissue is basically dead. Clear, bache? Right? So tracheids are the primitive water conducting elements of the xylem. What are they? Right? They are the primitive water conducting elements primitive water conducting elements of xylem. Right, water conducting elements of xylem. So when you talk about the structure here in the case of tracheids, so they are elongated or tube-like. What are they? They are elongated or tube-like structures. What are they? They are elongated or tube-like structures. And of course, you know that they are dead, means without protoplast. Without protoplast, cell walls are lignified there. Cell walls are lignified there. Yes or no? Yes, bacho. Yes or no? So elongated tube-like structures they are, bache. Cell walls are thick and lignified. And of course, if you talk about the lumen is going to be narrow or wide? Please tell me. What about the lumen? Lumen will be narrow or wide? Yes, students. You can even take the help from NCERT. What type of lumen will be there? Imagine cell walls are thick. And I just told you, narrow cavity, narrow spaces are there. Right? That is why that capillary action will be there. So obviously lumen is going to be narrow. Fine. It will be narrow. So elongated or tube-like structures. And it is tapering at either ends. Dead without protoplast, cell wall is lignified, and the lumen is going to be narrow. These are the tracheids, bache. Clear, bache? These are the tracheids. Fine. These are the tracheids. Okay. So even tracheids are unicellular. They are not multicellular structure. They are unicellular, made up of single cell only. This point is important. Tracheids are unicellular. And when you talk about their ends, just let's say, wait, I'll show you the diagram first. This diagram is from NCERT, na? So just see this. Tracheids, the ends are tapering. They are not pointed, they are tapering ends. And you will see, right, when you will see their ends, you will find the perforations there. Like pore-like structures will be there. It is perforated. So perforations are there. Right? Okay, perforations are there. Ends are perforated. Clear? Ends are perforated. So just have a look. So xylem is the main conducting tissue, bache. It will also provide the mechanical strength to the plant. Right? So when you talk about the tracheids, they are elongated or tube-like cells with thick and lignified walls and tapering ends. They are dead without protoplasm. Inner layers are having the thickenings which vary in the forms. And you know that in flowering plants, xylem and phloem are the main water transporting elements. That's all. Okay, so first of all, tell me any doubt here, and do include these points in your notes, right? Even you can write down on the NCERT itself. Fine. You can even mention these points on the NCERT itself. Okay. Fine. Sure. This is about the tracheids. This is about the tracheids. So inner layer of cell walls have thickenings which vary in form. That point is important. Okay, that point is important. Done. So pits are also present. Right? Pits are also be there. Done. So pits are present. Pits are present at the end walls of the tracheids. But now there is one more point. Do you know what is it? Just say now in the woods of gymnosperms, and pteridophytes, in the woods of gymnosperms and pteridophytes, walls contain, right? Walls contain tracheids. Basically, vessels are absent there. Do you know that? Vessels are absent there. So let me explain this part. This is important, right? So now after tracheids, what I just said, vessels are present. as a tertiary elements, as a water conducting channel, right? We have the tracheids, then we have the vessels. We have the tracheids, then we have the vessels. But presence of vessels is an advanced feature. Do you know that? Presence of vessels is an advanced feature. Okay, it is an advanced feature. Vessels are present in angiosperms only. Do you know that? They are present in angiosperms only. It is an advanced feature. They are present in angiosperms only. Now, I will explain one more thing. जैसे देखो, we have studied the plant kingdom. Yes, बच्चे, we have already completed the plant kingdom. Now you know that in plant kingdom we have algae, we have bryophyte, and then we have pteridophytes, the gymnosperms, and the angiosperms. Yes or no? In the case of plants, you know that we have algae, we have bryophyte, pteridophyte, gymnosperms, angiosperms. Pteridophyte, gymnosperms, angiosperms. That's what we have. So now when you talk about these pteridophyte, gymnosperms, and angiosperms, so for the first time, the xylem and phloem, they will appear in the pteridophytes. For the first time, we will see the vascular bundles in the case of pteridophytes. So pteridophytes, gymnosperms, and angiosperms, they are considered as the tracheophytes. Right? They are considered as what? They are considered as the tracheophytes. Now, what is the meaning of tracheophytes? The meaning of tracheophytes is, right? What is the meaning of tracheophytes? The meaning of tracheophyte is that they are having vascular bundle. The plants having vascular bundles. Right? They are the plants having vascular bundles. They are the tracheophytes. So, you have pteridophytes, gymnosperms, and the angiosperms. Pteridophytes, gymnosperms, and the angiosperms. So they are the tracheophytes. Now, the presence of vessels, it's a specific and it is an advanced feature of the angiosperms. Your gymnosperms and pteridophytes, they do not contain the vessels. But yes, again, we have few exceptions. Okay, what do we have? We have few exceptions. Jैसे in gymnosperms, 1 minute. Gymnosperms of order Neetae. Gymnosperms of order Neetales. Order Neetales. Like, you know, we have different orders. You know classification, right? Division, class, order, family, genus, species. So gymnosperms of order Neetales. Order Neetales mein you have the example of Neetum. Neetum is the genus. Right? You have the example of, yes, I'm talking about the gymnosperms. So I am talking about the exceptions here. So I am saying that vessels, it is an advanced feature, right? The presence of vessels, what is it? It is an advanced feature. It is present in the angiosperms only. It is not there in the monocots, right? It is not there in the monocots. And in some gymnosperms, there is an exception. Gymnosperms of order Neetales, which include your Neetum, which include your Ephedra, which includes your Welwitschia. It's Welwitschia, not Wolffia. Do not confuse it. Right. So, gymnosperms of order Neetales. Neetum, Ephedra, Welwitschia. Neetum, Ephedra, Welwitschia. And even in some pteridophytes. And even in some pteridophytes. Can you name that pteridophytes? That is a very common example. Can you name that pteridophyte? These are the exceptions here. So gymnosperms of order Neetales, Neetum, Ephedra, Welwitschia, and even in some pteridophytes, like you can say that Selaginella, Adiantum, but they contain the vessels. Okay, they are going to contain the vessels. So Selaginella, Adiantum, these are the example of your pteridophytes, and gymnosperms mein Neetum, Ephedra, Welwitschia, right? They contain vessels. Are you getting it? They contain vessels. Is it clear? Yes or no? Is it clear? These exceptions are important. Yes, bache? These exceptions are important. Done? Right? So pteridophyte, gymnosperms, angiosperms, they are the tracheophytes, means they are the plants having vesicular bundle. Okay, but presence of angiosperm, presence of vessel is a unique feature of angiosperms. But again, we have few exceptions. So gymnosperms of order Nettles, and even in some pteridophytes like your Selaginella, Adiantum, even one more example of Dryoteris is also there. Dryoteris is also there. So they contain what? They contain the vessels. Clear, bache? They contain what? They contain the vessels. This is what you need to keep in your mind. So when I talk about the vessels, if I talk about their structures, right? So vessels are also, bache, long tube-like structures, right? If I talk about the structure of the vessels, again, it is also a tube-like structure made up of many cells. So I told you na, tracheids are unicellular, but here it is not. It is tube-like structure made up of many cells, and that cells are known as vessel members. Okay, that cells are known as vessel members. So tube-like cells made up of many cells, and you will see in the vessels that ends are having, ends are also having the perforations. This is what we are going to see here. Okay, ends are also having the perforation. So they are long tube-like made up of many cells called vessel members, and obviously, right, here you will see that these vessel members are also interconnected. Okay, these vessel members are also interconnected. Fine, they are also interconnected through perforations, means through pores. Fine, and rest you know that vessels are also thick walled, thick lignified walls are there, right? They are dead, thick, without protoplast. Done? Yes? Any doubt? So here we have the diagram, vessels. So you guys can see the perforation. So vessels are multicellular, tracheids are unicellular. Right? Vessels are multicellular, tracheids are unicellular. Clear, bache? So see this. Let's read it from NCERT. So dekho, gymnosperms, they lack vessels in their xylem. Right? So here you guys can even mark that exceptions. Fine, bache, you can even mark that exceptions. And vessels is long cylindrical tube-like made up of many cells called vessel members, each with lignified walls and a large central cavity, and it is devoid of protoplasm. Clear, bache? Devoid of protoplasm means without protoplasm. So vessel members are interconnected through perforations in their common wall. So presence of vessel is a unique character of angiosperms. That's all, that's it. Any doubt? Yes, any doubt here? Done, bache? Sure. Next is the xylem fiber, basically, which is wood. Okay. It is basically wood. Xylem fibers are also known as wood fibers. But before that, tell me, any doubt? Sure, any doubt? Sure? Yes, but I just forgot to tell you this. Yes, even note down that in some angiosperms, right? In some angiosperms, vessels are absent. Do you know that? Vessels are absent, and you know that example, right? Example pe we have yucca plant. You know now we talk about the yucca and the pronuba moth. Yucca and the pronuba moth in the sexual reproduction in flowering plants. So that is the example. And other than that, Dracaena is also the example. Right? Dracaena is also the example. So these are the angiosperms in which vessels are absent. Clear, bache? These are the angiosperms where vessels are absent. Now the next part is your xylem fibers. So when you talk about the xylem fibers, so obviously their main role is to provide the mechanical strength. When you talk about the xylem fibers, right, they are also known as wood fibers, bache. What are they? They are also known as wood fibers. Majorly you will see them in the case of wood. And let me tell you, bache, they can be septate. Septa can be there. You know na, septa or aseptate. It can be septate or it can be aseptate. Xylem fibers. Clear? It can be septate or it can be aseptate. Again, here also the walls are going to be highly thickened and obliterated central lumens are there. Right? That lumen is not much basically. Okay? Obliterated central lumen is there. Done, bache? So they may be septate or they may be or without septa. That is aseptate. Okay? Done? Sure? So it is going to provide the strength. Okay, it is going to provide the strength. Done? So that's all. This is what we need to know about these xylem fibers. Highly thickened. Obliterated means very less. Right? Right? Central lumen is there. Lumen is not that wide. It is basically kind of destroyed. So it may be either septate or aseptate. This point is important. So they are also known as the wood fibers. And let me tell you, in secondary wood, right, in secondary xylem, you will see maximum xylem fibers are there in secondary xylem. Okay, maximum xylem fibers are there in secondary xylem. Clear, bache? Right, maximum xylem fibers are there in secondary xylem. That's important. And do you know, in the case of gymnosperms, their wood is very soft. Yes, gymnosperms mein na, that is gymnosperms wood is a soft wood. Gymnosperms wood is a soft wood. Fine. What is it? It's a soft wood. And that soft wood is without xylem fibers. Okay. It is without xylem fibers. Clear, bache? Clear, clear, clear. So next is xylem parenchyma. So parenchyma, it is obviously it is living. So cells are living and thin-walled here. So they are made up of cellulose. So their main function is going to be the storage. That's all. Fine. So how this xylem parenchyma is going to help? Its main function is of the storage. Right? Its main function is of the storage. Clear, bache? Done? It is what? It is storage. Okay. So storage of substance will be there. Cells are living, of course. So that's xylem parenchyma. Okay. That's xylem parenchyma. Jaise here it is written na, the radial conduction of water takes place by ray parenchymatous cell. Right, it is given a radial conduction of water is takes place by the ray parenchymatous cells. So, bache, we will talk about the dicot stem, the section of dicot stem. So, when we will see the section of dicot stem, you will see the space in between the vascular bundle. It is actually the medullary rays. Right, here you will see the medullary rays. So, here we see, right, this part is basically for the radial conduction of water and minerals. Okay. Okay. So this is what you have to see. The radial conduction of water takes place by the ray parenchymatous cells. So I will explain this to you. Done? Done, done, done. Any doubt? Sure? Any doubt? Clear? Which Hindi word I have used? Please tell me, Vedant. Okay, so just keep it in your mind. Two vascular bundles. In between these two vascular bundles, you have the parenchymatous cells. These cells are basically making that medullary rays or parenchymatous rays. They are helping in the radial conduction of the water. So once I will start the sections, you will understand it in a better way. Done, bache? So this is all about the xylem. Now, bache, in the xylem, you know, we have a few other terms also that we need to understand. Just say like xylem. We have two terms here. One is your protoxylem and another one is your metaxylem. Right? One is your protoxylem and another one is your metaxylem. Do you know the meaning of these protoxylem and the metaxylem? No doubt we will also talk about the primary xylem and the secondary xylem. Primary xylem which forms during the primary growth, right? Secondary xylem which will form during the secondary growth, right? But we are also having two words, that is protoxylem and the metaxylem. Protoxylem and the metaxylem. P for proto, right? Proto, the xylem that forms first. The xylem that forms first, it is the protoxylem, right? It is the protoxylem. The xylem that form later means after protoxylem, the xylem that will form is your metaxylem. It's simple, it's easy, isn't it? It's simple, it's easy. See, primary xylem is of two types. Primary xylem means which form during the primary growth, proto and meta. The first formed primary xylem, the xylem which will form first, that is going to be your protoxylem, and the xylem that will form later is your metaxylem. Clear bache? It is your metaxylem. So here you guys can see xylem mein protoxylem and metaxylem, that's what we have, and these two are the types of the primary xylem. So what is the primary xylem? That forms during the primary growth of the plant bache. It forms during the primary growth of the plant. Clear? Now bache, how that xylem is placed, okay, it is also going to create a difference. In some sections, let's say you can consider any transverse section, right? You can take the transverse section of root, you can take the transverse section of shoot, it can be anything. Actually, what's the point here? See, if your protoxylem, the xylem which will form first, metaxylem that will form after the protoxylem. If your protoxylem is facing, protoxylem is going to be small in size in comparison to the metaxylem. Like if I have to draw it here, right? See, protoxylem means cells are going to be comparatively smaller, and when I talk about the metaxylem, the cells will be comparatively larger. So if your protoxylem is facing the center, whenever you take the cross section of any stem, you take the cross section of any you take the cross section of any stem, you take the cross section of any root, right? Cross section means the transverse section. Then when you check the section under the microscope, you will look for the position of the xylem as well. And let me tell you, the position of the xylem can also tell that whether that section is of a root or it is of a stem, right? With the help of this position, we can also say that whether it is a root or it is a stem. So when your protoxylem, it is facing, like now this is a transverse section, let's say this is a transverse section, here you will see the epidermis, then hypodermis, cortex, whatever it is, here the endodermis, the pericycle, and here in between you will see the xylem. Okay, so if your protoxylem is facing the center, this portion is going to be the center, this part is going to be the center, and for the center we also use the word pith. What is the word that we use for the center? We use the word pith. What is the word bache that we use for the center? We use the word pith. So if protoxylem faces pith, mane center, then that arrangement of xylem is end-dark, right? It is end-dark, inside, end-dark, inside, end-dark, guys, inside, end-dark. So you will find such arrangement in the case of stem. Fine, in the case of stem. And now if protoxylem is facing periphery, if it is facing periphery like this, so if it is facing periphery, the outer side, right, then the arrangement of xylem is exarch. outer side, and you will see such arrangement in the case of roots. So now tell me, by looking at the section, by looking at the xylem arrangement, can we say which one is root, which one is stem? Of course. If it is a stem, end arc arrangement will be there. If it is a root, exarch arrangement will be there, right? If it is a stem, end arc. If it is a root, exarch arrangement will be there, right? So now what you see, now protoxylem, the xylem which will form first. Let's say this is the protoxylem that we have here. And then metaxylem, right? Then this is the metaxylem. So if this protoxylem is facing, if this protoxylem is in the pith region, if it is facing the pith, and if your metaxylem is facing the periphery, so can I say that the formation of xylem has started from the center? Can I say that the formation of xylem has started from the center? In the end arc arrangement, that's what I'm saying, na? Yes, bacho, that's what I'm saying, na, in the end arc arrangement, that it is the xylem which is facing the center. It is the xylem which is facing the center, like a protoxylem which is facing the center. It means the development of the formation of xylem has started from the center, and from center it has started moving towards the periphery. So if something is moving from center to the periphery, don't you think this is the word that we are going to use? Centrifugal arrangement will be there. Centrifugal arrangement will be there. Yes, tell me. It is the word that we are going to use, yes or no? Hanji, yes or no? Yes, bachcho, yes or no? है ना? So that xylem will be the centrifugal xylem. And when the arrangement is like this, your protoxylem is facing the periphery, it is facing the outer boundary, okay? In that case, this arrangement is exarch. Now it means the development of xylem has started from the periphery. So from periphery towards the center. Here, from center towards the periphery, right? From center towards the periphery. And here, from periphery towards center. So here the arrangement is exarch and it is centripetal xylem. This one is in the case of stem, in the case of root, right? And I think you all can observe the difference here also, that it is the protoxylem. So if it is a protoxylem, bachcho, right, cells are small in size, hai na? Cells are smaller in size and they are having tertiary elements, no, the tracheids and xylem parenchyma. You know, in protoxylem, you will only find tracheids and xylem parenchyma. Do you know that? That's extra. If you want to note down, okay, otherwise you can leave it as well. Okay, you can even leave it. Yes, no, not the tracheids, it is the tertiary elements. So in the protoxylem, you will see tertiary elements, means tracheids and vessel, and even you will find xylem parenchyma. Now when it is the metaxylem, cells are bigger in size, so here you will find tertiary elements. Okay, you will even find xylem parenchyma, you will even find xylem fibers. So that's how it grows. Clear? So this arrangement is very important and it will also help us to check the differences in a root and in a stem, right? So end arc, exarch. Any doubt? Any doubt? Tell me, bache. Then we will start the phloem. If there is any doubt here, do let me know. Then we will start the, right? After I'll clear your doubt and then we will start the phloem. Okay. So see this diagram of the phloem cells. Just look at it. Sieve plate, sieve tube, phloem parenchyma, companion cells are there. See the diagram from NCERT. Right? Sieve pore, sieve tube elements, phloem parenchyma, companion cells are present there. And bache, see this diagram. right? See how the xylem is also arranged. Xylem, parenchyma, vessels, and the tracheids. Okay, xylem, parenchyma, vessels, and the tracheids. So that's how they are arranged. So diagrams are also important, right? Whenever you revise the NCERT, whenever you read anything from the NCERT, always check the descriptions of the description of the diagram as well. Fine, clear, sure. Okay. So see this perforation plates in the vessel members, that is how they are linked. Tracheids, they are, right? Fibers, they are. These are the phloem. In the case of phloem, look at the sieve plates, right? Why do we use the word sieve here? Sieve, it is having the pores, obviously for the smooth flow of the things, and sieve cells. Along with that, companion cells are also present. And see this sieve area. Sieve in Hindi is channi, right? Jaise we basically with the help of these sieve plate, the food will be transported. Okay, the food will be transported. Done? So now what we have to start? We need to start the phloem. So if there is any doubt, please, again I'm asking, do let me know. Do let me know if there is any doubt. Sure? Okay, after phloem I'll give you the break. Okay? Don't worry, I'll give you the break, and then after that we can talk about the sections. And trust me, sections are not difficult. See, if you will understand the ground tissue, if you will understand how the tissues are arranged, then you just need to draw a comparison that in dicot stem it is like this, in dicot root it is like that. That's all. Okay, that's all. Fine. So let's talk about the phloem. Okay, let's talk about the phloem. So we know that phloem is the main food conducting tissue, isn't it? It is the main food conducting tissue, and the flow of things is flow here is bidirectional. So phloem is made up of, yes, sieve tubes, companion cells, phloem parenchyma, and phloem fibers. So these three are living, and this one is dead. Any doubt? So if it is dead, you know that what is the role of this phloem fiber here? It is meant for the protection. Clear, bache? It is meant for the protection here. So let's start with the sieve tubes. Okay, let's start with the sieve tubes first. Fine? Sure? Okay, just a minute. See, bache, we have two words here. One is the sieve cells, another word is the sieve tube, or you can also call it as sieve tube elements. It says one and the same thing. Now, bache, when you use the word sieve cells, right, it is a primitive feature. Of course, it will be there in the gymnosperms and the pteridophytes. Right, it is a primitive feature. It will be there in the gymnosperms and the pteridophytes. And when we say sieve tube element, so it will be there in the angiosperms. Okay, it will be there in the angiosperms. Let's imagine now that sieve cell has formed a tube here. Right, it is a tube here. Are you getting my point? It is a tube here. Are you getting my point? So sieve tube element is the feature of angiosperms, where sieve cells, they all are joined and they have formed a tube-like structure. Clear, bache? They have formed a tube-like structure. So here, this is what you guys can write down, that all the sieve elements, they are joined and they have formed a tube-like structure. So the ends here, ends of these sieve elements, they will contain the perforations. What will you see here? You will see the perforations here, the pore-like structures here. And that is what, that is why it is acting like a complete, right? It is basically a complete tube-like structure. Any doubt? Yes, bache. Any doubt? So end walls are basically perforated. They are the sieve pores. Okay. They are the sieve pores. Clear, bache? Yes or no? Yes or no? Sure. So these perforations, these are basically the sewage, and they are associated with what? Here, companion cells are present. What will you see here? You will see the companion cells here. Companion. Bache, the meaning of companion, you know the meaning of companion, isn't it? Isn't it? So that is why here also, because these cells are associated with these sieve elements, that is why we call them as companion cells. Now let me tell you, bache, companion cells are also absent in your gymnosperms. Okay, and even in your pteridophytes. Fine, companion cells are absent in gymnosperms, right, bache? They are only present in the angiosperms. So in gymnosperms, we have albuminous cells, right? At the place of these companion cells, albuminous cells are present. Clear? Albuminous cells are present. Clear, bache? So companion cells are absent in gymnosperms. In gymnosperms, albuminous cells are present. Companion cells are absent there. Now, bache, when you talk about these sieve tube elements, we compare them with the mature, right? We compare them with mature human RBC. Do you know why? We compare it with the mature human RBCs, right? With mature human RBCs. Clear? Do you know why? Yes. Because at maturity, they become enucleated. Okay. So that is why we compare it with human RBC. Just say human RBC may, in the growing phases, in the initial stages, you will find the nucleus, but later on, when the RBCs, they attain maturity, the nucleus will not be there. They will become enucleated. Same is the case here. When you talk about the sieve tube elements at maturity, when they attain maturity, right, they become enucleated. Right? When they attain maturity, they become enucleated. Clear? Is it clear? Yes or no? Yes or no? So now the question here is that if they do not have the RBC, then who's going to control their function? Companion cell. The nucleus of companion cell will control the function of the sieve tube element. Okay, that is important. The nucleus of the companion cell is going to control the function of the sieve tube element. Is it clear? Yes, bache, is it clear? Yes or no? Quick, yes or no? Done? So that's important. Okay, that's important. So initially they were living, but when they are mature, they will lack nucleus. Okay, so even sieve cell, even sieve tube elements, initially they were living, but later on when they will become mature, they lack the nucleus. So if there is any doubt, please tell me. Okay, so companion cells, again, they are the special parenchymatous cells having their nucleus. Their nucleus is going to take over the function of these sieve tube elements. That's all. This is what you need to remember. Clear, bache? This is what you need to remember. Okay. Sure? Sure, bache? Done? Okay, the next part. Yes, the next part. Yes, albuminous cells. Okay, so next is your phloem fiber. So I told you already about the phloem fiber, isn't it? I told you already about the phloem fiber. So when we use the term phloem fibers, they are also known as vast fibers, right, bache? And they are dead. Okay, so here they are dead, they are without protoplast. What are they? They are dead, they are without protoplast. So if they are dead and if they are without protoplast, so you know that, right, bache? You know it very well. See, phloem fibers, bast fibers, they are dead, they are without protoplast. Exactly, right? They are dead, they are without protoplast. So it will provide the mechanical strength, right? What will it do? It will provide the mechanical strength to the phloem, right? It will provide the Yes, everyone. Mechanical strength to the food. Clear, bache? Sure? Yes, it is going to provide the strength. That's all. Okay, that's all. Phloem fibers. Okay, phloem fibers. Okay, so yes, and one more point is, bache, they are absent. Phloem fibers are absent in primary phloem, right? The phloem which will form first, you will not find these phloem fibers there, but they are present in secondary phloem, right? They are present in secondary phloem. Now, bache, the word is, okay, the word here is fiber. So I don't think that I need to tell you the structure. I hope you remember that structure is going to be pointed. It is like this. Okay? The structure is going to be pointed. I have already explained it. That is how the fibers are present. Okay. So next is what? Next is what? Phloem parenchyma. Okay. Next is what? Next is the phloem parenchyma. So you know the role of phloem parenchyma, isn't it? You know the role of phloem parenchyma. So if it is phloem parenchyma, so of course cells are living, they are thin walled. Fine, cells are living, they are thin walled. Yes or no? I'm repeating this again. Cells are living, they are thin walled. They are for the storage of food. Isn't it? They are for the storage of food. Fine. And bache, this phloem parenchyma, please note down, that is important. It is absent in monocots. That's important. It is absent in monocots. It is absent in monocots. Clear, bache? Phloem parenchyma. So it is meant for the storage of food, storage of resin and all. Okay? So it is absent in the monocots. That's the important point. And obviously it will also help in the water conduction. Okay? It will also help in the water conduction. Done? That we will see in the secondary growth. So now let's read it from the NCERT, and this point is important. Please mark it. This point is important. So here we can see, bache. So phloem transports food material from leaves to other parts of the plant. So phloem in angiosperm is having sieve tube element. So this is sieve tube element, not the sieve cell. Companion cells, phloem parenchyma and phloem fiber. Gymnosperms mein, they have the albuminous cells. And yes, one more point, bache, albuminous cells are also known as Starsberger cell. है ना? देखो Starsberger. It is not Starbucks. It is Starsberger cell. Right? Albuminous cells are also known as Starsberger cell. Clear? Clear, bache? So gymnosperms, they lack sieve tube, they lack companion cell. Right? So sieve tube elements are nothing. They are the tube-like structure arranged longitudinally and are associated with the companion cells. Clear, bache? So they are end walls. In the case of sieve tube, they are perforated in a sieve-like manner. I told you already. So mature sieve element possesses a peripheral cytoplasm and large vacuole but lacks a nucleus. Very important line. Okay, this line is very important. Clear? This line is very important. So the function of sieve tubes are controlled by nucleus of companion cell. And bache, in the plant physiology, we will talk about the mass flow hypothesis, bulk flow hypothesis. So you will learn that how these companion cells are helping in maintaining a gradient in the sieve tube cells, right? So it is actually helpful there in maintaining the gradient there. Fine, bache. So next part is, you can see here the companion cells are specialized parenchymatous cells. Companion cells are what? They are the specialized parenchymatous cells which are closely associated with the sieve tube elements. Clear? So they are connected by pits, by pit field present between two common walls. Right? Just suppose karo, this is one wall, this is one room, this is another room. There is a common door in between. Same is the case here. In between your sieve tube cells and the companion cells, you will see a common pit field will be there. Pits are present there. Okay, pits are present there. So companion cells help in maintaining the pressure gradient in sieve tubes. That we will learn in that bulk flow hypothesis. So as I said, phloem parenchyma, which is having elongated tapering cylindrical cells having dense cytoplasm. Okay, so phloem parenchyma mein bache, the wall is composed of cellulose and has pits through which plasmodesmatal connections exist between the cells. That's obvious. Parenchyma it is, plasmodesmatal connections are there. Okay, plasmodesmatal connections are there. So phloem parenchyma stores food material, other substances, right? So it is absent in most of the monocots. So that point is very important. Okay, it is very important. Just I told you, collenchyma, it is absent in roots and monocots. Phloem parenchyma absent in monocots, right? So please make a copy, and in which you can mention all that points. So you should note down all that points. So phloem fibers, also known as vast fibers, again bache, they are fibers. Sclerenchyma is dead cells they are. So they are generally absent in primary phloem but are present in secondary phloem. Clear? So they are much elongated, unbranched, have pointed needle-like apices, means needle-like tips are there. Needle-like tips are there. Okay, so cell wall of phloem fiber is thick. At maturity, they lose their cytoplasm, become dead. So phloem fibers of jute, flax, hemp are used commercially, told you already. And here also, bache, protophylum and metaphylum is having the same meaning. Okay, protophylum and metaphylum is having the same meaning. Protophylum, which will form first, metaphylum, which will form later. So protophylum mein narrow sieve tubes are there, metaphylum mein larger sieve tubes are there. So that is all about the phloem bache, right? That is all about the phloem. The next that we need to start is the ground tissue, right? Next we need to start is the tissue system basically. Okay, what we need to start? We need to start the tissue system, the last topic for today. After it, we will discuss sections. Sections are very easy. If you will understand tissue system, then there will be no problem at all in sections. Right? Okay. So I will give you a break. Now it's 9:10. Basically, it's 9:07. You consider it as 9:10. So 9:10 to 9:30. Break time. Okay, that will be your break time. Let's finish it in one go, bache. So 9:30 to 10:30 class. Fine. And we will finish this tissue system wala part. We will finish the sections then. Okay, we will finish the sections then. Done? Done? So let's finish it in one go. Okay, let's finish it in one go. And Sunday at 1 p.m. we will talk about the secondary growth. And you will get the time for the revision as well. Sunday 1 p.m. secondary growth will be there. Okay, so take care. So I'll meet you after 20 minutes. Hi students. Hello. Hello, hello, hello. I'm sorry for being late. Actually, I was having my dinner. Right? Actually, I was having my dinner and that's why I got late. I'm very sorry for that, bacho. I tried not to, but again, na, some things happened always. Chaliye. So the next part is very easy, and I think we all are charged up, isn't it? Hannah, we all are charged up, isn't it? Okay, so can we start? So quickly share the link with others so that they can also join the class. Quick, just share the link with others as well so they can join the class. Right, very less number of students are there. What happened? Now to you people should be, active and all. But I will take another 1 hour. I will take another 1 hour. Okay, Jasper is saying, what does organ mean in parenchyma? But organ mean in parenchyma is, see, parenchyma is a tissue. So it is going to make the plant organs. So when you talk about the soft organs of a plant, like in the fruit pulp also, you will find parenchyma. When you talk about the stem, the root, you will see the majority of the tissue is the parenchyma basically. That's the point. Okay, that's the point. So the topic now that we need to start is tissue system, and here in tissue system you will talk about the epidermal tissue system, and trust me bache, if you will master this part, okay, if you will master this part, then there will be no problem at all in the sections. Sections are going to be very easy for you. So we have the epidermal tissue system, we have the vascular tissue system, right, and then one more is there, ground tissue system. Okay, so we have vascular tissue system, ground tissue system, we have epidermal tissue system. That's how we divide the sections. Okay, that's how we divide the section. Let me quote one example. I hope you people in your schools you have performed some experiments, like whenever we used to take a thin section of a plant, right, we see some cells. So ultimately here in this tissue system we will see how that cells are arranged. Okay, how that cells are arranged in the dicot stems, in the dicot roots, in the monocot stem, in the monocot roots, and even in the case of leaf. So basically we are dividing that tissue system into 3 parts. The first one is the epidermal tissue system, the second one is the vascular tissue system, the third one is the ground tissue system. Okay, the third one is the ground tissue system. This is what we have. Are you getting my point? This is what we have: epidermal, vascular and ground. Epidermal, vascular and ground. Right? Repeat it with me: epidermal, vascular and ground. Now when you talk about the epidermal tissue system, here you will talk about epidermis. You are going to talk about the stomata here. Okay, and even you are going to talk about the epidermal appendages. That's what we need to discuss here. Okay, that's what we need to discuss here. And in the vascular tissue system, of course, you will talk about the xylem and the phloem. Right? You will talk about the xylem and the phloem. Yes or no? What are we going to discuss here? We are going to discuss about the xylem and the phloem. And when we talk about the ground tissue system, are you getting my point? When we talk about the ground tissue system, here in ground tissue system, all tissues, right? All tissues except epidermal tissue system and vascular tissue system, it comes under the ground tissue system. Like remove epidermis part, epidermal tissue system part, remove vascular bundle wala part, whatever is remaining, that comes under the ground tissue system. Okay, that comes under what? That comes under the ground tissue system. This is what we need to understand. Okay, right, that is what we need to understand. Is that clear? Yes, bacho. Is that clear? Sure? Sure. Okay, so let's start with the epidermal tissue system. Let's start with what? Let's start with the epidermal tissue system. So here you know that epidermis is the outer protective covering. Whenever we use the word epidermis, okay? See, epidermis is the outer covering, right? So here, first of all, what we need to discuss, epidermis. And as I said, what is epidermis? It is the outer covering, outer protective covering it is. Yes or no? What is it, bache? It is the outer protective covering. Yes or no? That's the point. Tell me quickly. That's the point. It is the outer protective covering. It is the outer protective covering. Okay. And in the case of, and you know that in stems, this epidermis, it is the outer protective covering. Here you will find epidermal cells. What type of cells are they? They are the elongated cells, bache. They are the elongated cells. They are compactly arranged. So basically in the epidermis, right, in this layer, you will not see any intercellular spaces. So cells are what? Cells are compactly arranged here. Cells are what? Cells are compactly arranged here. So what do we have here? See, epidermis, outer protective covering, right? Epidermal cells, what kind of cells they are? They are the elongated cells, bache. They are the elongated cells. And how are they arranged? They are compactly arranged, right? How are they arranged? They are compactly arranged. Is that clear? Is that clear? Yes or no? Yes or no? They are compactly arranged, means there is no intercellular space. Let me write down. Okay, there is no intercellular space here. That is what we need to take care. Now in stems, your epidermis is covered with, okay, it is covered with a waxy cuticle. It is covered with what? It is covered with waxy cuticle. Like cuticle is covering the epidermis. So clear? And so that you know water loss will be less. That is why water loss will be less. But in roots, fine bache, but in roots you know that what is the scenario. In roots, epidermal cells are not covered with, yes, epidermal cells are not, they are not covered with cuticle. So in the case of roots, which term are you going to use for it? Tell me, which term will you use for it? In roots, epidermal cells are not covered with cuticle. So here the term that we will use is epiblemma, right? The term that we will use is epiblemma. What is it? It is epiblemma. Is it clear? Okay, is it clear? You can see this. So epidermis, when it is covered with the cuticle. So epiblemma, it is the epidermis of the root which is not covered with the cuticle. Any doubt? Yes? Any doubt? Next part is the epidermal appendages. Okay, so these are basically the outgrowths of the epidermal cells. What are they? They are basically the outgrowths of epidermal cells. Yes or no? What are they? They are the outgrowth of epidermal cells. So, bache, here we have two things. One is root hair. Right. Another will be trichome. One is the root hair. Another is the trichome. One is what? Root hair. Another is the trichome. Obviously, you know that root hairs, right, present in the roots, of course, and they are meant for the absorption of water and mineral. So always remember your root hairs are going to be unicellular. What are these root hairs? They are unicellular. Now when you are talking about the trichomes, they are also the epidermal appendages. They are present in stem. They are not present in root, bache. They are present in stem. So they are usually multicellular. They are what? They are usually multicellular. Are you getting my point? They are usually multicellular. So have a look again, bache. So see, what do we have here? We have the epidermal appendages. Okay, so outgrowths of epidermal cells. So root here and trichomes. Root here and trichomes. What do we have here? Root here and trichome. So root here may they are unicellular. Trichomes, they are present in stem. They are usually multicellular. Okay, so what is the function of root hairs, bache? Root hair are meant for the absorption of water and minerals. Right, they are basically for the absorption of water and minerals. Are you getting my point? For the absorption of water and minerals. And here, when you talk about the trichomes, okay, when you talk about the trichomes present in stem, usually multicellular, they prevent the water reduction, right? They prevent the water loss basically. They prevent the water loss from the stem. Is it clear? Yes or no? Is it clear? So epidermis, elongated, compactly arranged cell, right, bache? They are parenchymatous. Okay, so they have two appendages, trichomes and root here. So trichomes are usually multicellular, present in shoot system, bache. So what is their function? They help in the reduction of water loss, right? So they prevent the water loss. This is what you guys can write. Okay. Okay. Sure. Sure, bacho. So these trichomes, they can be branched also. Okay, they can be branched also. And root hairs, I told you, root hair cells, I told you already about it. Done, bache. So this is the epidermis. Always remember, epidermis is also made up of parenchymatous cells. They are also made up of what? They are also made up of parenchymatous cells. Is it clear? Yes. It is made up of what? It is made up of parenchymatous cells. Any doubt? Yes, bacho. Any doubt here? So root here, trichomes, this is what we have. This is what we have here. Clear, bache? This is what we have here. Done? Done. Sure? Is it clear? So even you know that I just told you that you will also find stomata. Right, you're also going to see the stomata in the epidermal tissue system. So what is stomata? Right, you know that, but you see, it's a very basic topic. So in the epidermis, some cells, they will become modified. Okay, some cells, they will become modified. Are you getting it? In the epidermis, some cells, they will get modified. Okay, so here you guys can see that modified cells are basically your guard cells. So that guard cells are covering one opening, stomata, and the singular here is stoma. Okay, singular here is what? Singular here is stoma. So singular here is stoma. So you know that stomata is for the plural, stoma means singular. So here this is a pore-like structure. What is it? It is a pore-like structure. Yes, bacho, what is it? It's a pore-like structure. It is a pore-like structure by which gaseous exchange will be there. Okay, it is a pore from where the gaseous exchange will be there, and there will be the loss of water in the form of vapors. There will be the loss of water in the form of vapors. That is what stomata is, and it is covered with these guard cells. So these guard cells are also the modified epidermal cells, and moreover, bache, these guard cells are further covered with, you know, they are further covered with other epidermal cells. They are also known as subsidiary cells, or you can also call them as accessory cells. Right, you can also call them as accessory cells. So what do we have here? Subsidiary cells and the accessory cells. Subsidiary cells and the accessory cells. Is that clear? Right, so here you guys can see that these guard cells are also having the chloroplast. So it means they will also assimilate carbon dioxide. They are also going to do the photosynthesis. Okay, bache? Okay, so this complete structure is basically the stomatal apparatus. What is it? It is the stomatal apparatus. Now, what you need to remember here. Now, see. Yes, in dicots, in dicotyledons, right? in dicotyledons, what is the scenario? Here your guard cells are kidney shaped, right? What is going to happen here, bache? Here the guard cells are what? They are the kidney shaped. They are bean shaped or they are the kidney shaped. Are you getting my point? Yes. And in the case of monocots, what will you see here in the case of monocots? In the case of monocots, these guard cells are the dumbbell shaped. What are they? They are dumbbell shaped, right? They are dumbbell shaped. Fine, that's what you need to remember. It's an important question. Okay, done, bache. And here you will see, always remember in the case of guard cell, right, the inner wall will be thick. Inner wall is what? It is thick. And this wall is having radially arranged cellulose, cellulose microfibrils. What will you see here? You will see radially arranged cellulose microfibrils. Is that clear? Radially arranged cellulose microfibrils are present here. Done, bache? So whenever there is a change in the turgor pressure, like when your guard cells, they will become turgid, your stomata will open or else it will get closed, right? Or else it will get closed. So that is how it is arranged. So this is all about the epidermal tissue system. So you will see the epidermis here. You will see the epidermal appendages, include root hair and trichomes, and then you will see the stomata there. So this guard cell, this pore-like structure, stoma or stomata, and these subsidiary cell, together they are making stomatal apparatus. Fine. Together they are making stomatous apparatus. Clear, bache? Clear, bache? Sure? Any doubt? So this is the epidermal tissue system. Okay, this is the epidermal tissue system. Anji, any doubt? Sure? Are you sure? Fine. Chaliye. So one more thing I would like to mention here. This is sometimes in epidermis, na? Sometimes in the epidermis, like in monocots. In monocots may particularly your grasses. Fine, particularly your grasses. In monocots or particularly what? Particularly, yes, in monocots, particularly what? Particularly grasses. In grasses, na, in epidermis, you will find some motor cells, also known as bulliform cells. What will you find there? Motor cells, also known as bulliform cells. What are they, Akanksha? They are the motor cells, also known as bulliform cells. Isn't it? See there. In monocots, in grasses, what do we have? We have the motor cells, also known as bulliform cells. Motor cells, also known as bulliform cells. Now, what is the role of these motor cells? Just say, I think you have noticed that your grass leaves, right? Sometimes they show the curling, right? They show a kind of bending. Have you ever seen it? This is sometimes in the case of grasses. This is what we used to say. This is suppose sometimes the leaf surface is directly exposed. Okay, the leaf surface is directly exposed sometimes. This is what we observe. But sometimes what happens? There is a curling of the grass leaves. There is the curling of the grass leaves. Why is it so? When there is the water scarcity, in that scenario, what is going to happen, bache? When there is the water scarcity, in that scenario, what is going to happen? Right, the motor cells which are present in the epidermis, they will lose their water. Okay, motor cells present in motor cells, they will lose the water. What will they do? They will lose the water. Okay, so during water scarcity, because these motor cells, these bulliform cells, because they have lost their water, so the leaf surface will get folded. There will be the curling of the leaf surface. Are you getting it? There will be the curling of the leaf surface. Are you getting it? And now when the conditions will resume, when again they will get the water. In that scenario, what is going to happen? Motor cells, they will again become turgid. Again, the leaf surface will be exposed. Fine. Again, the leaf surface will be exposed. Are you getting it? Yes. Are you getting it, students? Any doubt? Any doubt? Okay. Sure, bache? Sure. Done. So the next thing that we need to discuss is your ground tissue system. What we need to discuss next? It's ground tissue system. It is the ground tissue system. Okay, so it is the major part of the tissue system. Majority of tissue system includes your ground tissue system. So after leaving your epidermal tissue system and vascular tissue system, what you guys are going to get? You will get the ground tissue system. Are you getting it? Yes. Are you getting it? Let me write down. All tissues except epidermal tissue and vascular tissue comes under ground tissue system. They comes under ground tissue system. Are you getting my point? They comes under what? They comes under ground tissue system. Are you getting it? Are you getting it? So ground tissue system mein what are you going to get? Yes bachcho, what are you going to get? Hypodermis, cortex, endodermis, pericycle, medullary rays. I will explain it, right? So do you know about this or not? Yes, that in the ground tissue system, we will be having what? We will be having the hypodermis, cortex, endodermis, pericycle, yes, pericycle and medullary rays, right? And the medullary rays. But this is what you need to remember, bache, because it is a PYQ, that all the tissues except your epidermal tissue system and the vascular tissue system comes under the ground tissue system. So basically it is the largest tissue system. The ground tissue is what? It is the largest tissue system. And students, please listen to me very carefully, because if you have mastered this particular part, the tissue system, then all the sections are going to be very easy for you. Okay, you just need to look at the diagram and then you need to write, then you need to tell this is dicot stem, this is dicot root, this is this, this is that. Trust me, it will be very easy for you then. Okay, it is going to be very easy for you then. So any doubt from the epidermal tissue system? I hope there is no doubt, because this part is very, very easy. Now, the ground tissue system, largest tissue system, all tissues except epidermal tissue system and vascular tissue system will come under this. Now let's divide it. Okay, so ground tissue system includes hypodermis, cortex, and in this sequence you have to write it: endodermis, pericycle and medullary rays. And medullary rays. Fine. Hypodermis, cortex, endodermis, pericycle, medullary rays. I am repeating this again. Hypodermis, cortex, endodermis. Yes, bache. Hypodermis, cortex, endodermis, pericycle, and the medullary rays. Actually, bache, how are we going to divide it further? Let me tell you. Right. We can also divide it further. Jaise, let's say in ground tissue system, we say majority of part includes cortex. Okay, we can also divide it. Jaise we used to say majority of part is of cortex, right? There are two ways to divide it. See, let me explain this again. In the ground tissue system, what do we have? We have hypodermis, we have the cortex. Yes, bacho, what do we have? We have the hypodermis, we have the cortex, we have the endodermis, right? The pericycle is also there. After the endodermis, what do we have here? We have the pericycle, and even the medullary rays are also there. Right, medullary rays are also there, and even pith pith is also there. In general, this is what we consider in the ground tissue system. What is there in the ground tissue system? Hypodermis, cortex, endodermis, pericycle, your medullary rays, and the pith is there, right? Let me show you the diagram. Then it will be easy for you people to remember this. जैसे ये देखो, let's say this is the part, ठीक है? Epidermis, okay, epidermis is there. This is the general cortex, endodermis, pericycle, right? This part, the central portion is basically the pith, and in between the vascular bundle, that space, right? Majorly we will see it in the case of dicot stem. See this part, right? Epidermis, outermost covering, below epidermis, hypodermis. This is the cortex part. Then comes endodermis, then comes pericycle. Here you have your vascular tissue that we will not consider under ground tissue. In between these vascular tissue, these are the medullary rays, again the part of the ground tissue, and this pith is also the part of the ground tissue, right? This pith is also the part of the ground tissue. Now in general, this is what we have when we talk about what? When we talk about the ground tissue system. Again, I'm telling you, if you will listen to me carefully, the topic is going to be very easy for you. That's a promise. Okay, that's a promise. So how do we divide it? This is in general. Now you are my NEET aspirants. How are you going to focus here? See, in GTS you will say majority of part is your hypodermis. Then comes your general cortex. Now under cortex, this is what you have to study, right? Means in ground tissue system, we are further dividing it. Okay, Shiva Kumar, we are further dividing it. We used to say that in the ground tissue system, we have the cortex. Let me write it here. We have the cortex, and this cortex will include your hypodermis. It will include your general cortex, and then it will include your endodermis. Now I will explain NCERT with the help of this. Okay, so what will be there in the ground tissue system? In the ground tissue system, we will say there is the cortex, and then pericycle, and then the pith. That is how it is explained properly, right? Because in this way, the NCERT will become easy for you. That's why I'm teaching you in this way. Okay, that's why I'm teaching you in this way, that GTS include cortex, and cortex will include hypodermis, general cortex, endodermis. Then other than cortex, we have pericycle, we have pith, right? Ultimately, they are the same points, right? Ultimately, they are the same points, but I am explaining it in a different way. Why? Because we have to make the NCERT easy. Okay, we have to make the NCERT easy. Okay, so see, GTS, largest tissue. All tissues except epidermal tissue system and vascular tissue system comes under GTS. In the GTS, majorly what are we considering? We are considering the cortex. We are considering what? We are considering the cortex. That cortex include hypodermis, general cortex, endodermis. And then we have the pericycle, and we have the pith. Fine, we have the pericycle and we have the pith. Now I hope you remember this. Open up your NCERT, there is one line written that it is the endodermis which is the innermost layer of cortex. It is the endodermis which is innermost layer of cortex. I hope you know about it. I told you na, NCERT will be easy, right? NCERT will be easy for you then. It is given in NCERT, yes or no, that it is the endodermis which is the innermost layer of the cortex. Yes, bache. Tell me. Please tell me, bacho. So see, in this way you can remember it easily. So GTS mein cortex is having hypodermis, general cortex, endodermis. Then pericycle and pith is written. So it is the endodermis which is the innermost layer of cortex. But we have to start with the hypodermis first. So hypodermis, it is a layer below epidermis. You know that in the case of dicot stem, it is it is collenchymatous. This hypodermis in the case of dicots, it is collenchymatous. And in the case of monocot stem, what is this? It is sclerenchymatous. This is what we have covered in Collenchyma and Sclerenchyma. I hope you remember, right? Just now we have completed this portion. Just now we have completed this portion, that in the case of dicot stem, you will see collenchymatous hypodermis. In the case of monocot stem, you will see sclerenchymatous hypodermis. And what is the role of hypodermis? It will provide the strength, right? It will provide the strength. Is it clear? Is it clear? So this hypodermis, what is the role of this hypodermis? It provides strength, bache, and you will only see hypodermis in the case of roots, not in the case of in the case of stem, not in the case of roots, right? So see, now imagine the root structure. In the root structure below epidermis, we are not going to say there is a hypodermis. We will directly say there is a general cortex. Clear, bache? We will directly say there is a general cortex. Got it? Got it? So I'm explaining it again. This is what we have in GTS, and here we are dividing it. We are saying cortex include hypodermis, general cortex, endodermis. Then comes pericycle and pith as the part of GTS. Cortex mein below epidermis there is hypodermis. Hypodermis is present only in stem, dicot stem, collenchymatous, monocot stem, sclerenchymatous. Okay, and it is going to provide us the strength. It is going to provide the strength, and it is present in stem only. In roots and in leaf you will not see hypodermis. Any doubt, students? Any doubt, students? Yes. Any doubt that you have? Sure, sure. So general cortex, it includes parenchymatous cells, and you know that what is the role of that cells? It is going to include what? It will include parenchymatous cells, and they are meant for the storage of food. They are meant for storage of food, right? They are meant for storage of food. Now I will add one more point here. GTS in plants, GTS, ground tissue system in plants is mesophyll. In the case of plants, right, that ground tissue system is nothing, it is the mesophyll. So here mesophyll cells are having chloroplast. Mesophyll cells will perform the photosynthesis. So in the case of leaves, I'm writing the plants here, sorry, it should be leaves. In leaves, GTS in leaves is mesophyll. In the internal structure of leaf, what will you see? That mesophyll cells are there. So mesophyll is nothing, it is the ground tissue. Mesophyll is nothing, it is the ground tissue, and you know that these mesophyll cells, they are having the chloroplast, they will perform the photosynthesis there. Clear? So in general, what is the role of general cortex? Having parenchymatous cell and going to store the food. Having parenchymatous cell and going to store the food. Any doubt? Yes? Any doubt? Sure? Sure, sure, sure. So next to it is endodermis. Next to it is endodermis. Now I need this space. So endodermis is the innermost layer of cortex. Endodermis is what? It is the innermost layer of cortex. Now bache, in roots, right, endodermal cells contain thickenings that are Casparian strips, right? Casparian strips, and that Casparian strips are water impermeable because they are suberized. They are what? They are water impermeable due to deposition of suberin. Due to deposition of suberin. Clear, bache? We have the Casparian strips in the case of roots. Let me show you. Just in the case of roots, endodermal cells, they are having some thickenings, right? Endodermal cells, they are having the thickenings. So what are that thickenings? They are the Casparian strips, right? Casparian strips. So as I said, these Casparian strips, they are water impermeable. Okay, they are water impermeable. They will not allow the water movement, bache, because they have the suberin deposition. Clear? They have the suberin deposition. Okay, Casparian strips. Now, when you compare it with the stem, stem में क्या होगा? Do stem contain Casparian strips? Yes, बच्चे. Do stem contains Casparian strip? Tell me. Do stem contain Casparian strip? Quick. Quickly tell me. Quick, quick, quick. So, bache, in the stem, you will see starch sheath. What will you see? You will see starch sheath. In stem, Casparian strips are absent in endodermis. Casparian strips are absent in endodermis. Okay, so here it is the starch sheath only, starch deposition is there. And let me tell you, bache, this is another important point. Endodermis is absent in monocot stem. It is absent in monocot stem. Bache, that's why I'm telling you, listen to me carefully, note down these points, then all the sections will be easy for you. So basically, in the case of dicot stem, you will see that endodermis is known as the starch sheath there, right? Endodermis is known as the starch sheath there. It is not having Casparian strips. Okay, right? And here in the roots, what will we see? That endodermal cells are having Casparian strips. These are the depositions of suberin basically. So they will form a system, a watertight system. Water will not be able to cross these Casparian strips. Clear, bache? Clear, bache? So dicot stem you will see endodermis, but that is actually the starch sheath. We are calling it as starch sheath. But in monocot stem, it is absent. Monocot stem, it is absent. Done? Sure? Sure, bache? Sure? And you know, in endodermis, this endodermis is also having, you know, some thin walled cell. It is not mandatory that all the Casparian strips and thick walled cells are there. Some thin walled cells are also there in that endodermis, and we used to call them as passage cells. I will explain it in sections. Right? When we will discuss the sections, then I will discuss it. So any doubt here, Deena? Yes, bacho. Any doubt here? So dicot stem is having endodermis, which is known as the starch sheath. Okay, which is known as the starch sheath. So next to it is the pericycle. Next to it is what? It is the pericycle. You know what, bache? Pericycle is also considered as the outer boundary of the vascular tissue. Right? It is also considered as the outer boundary of vascular tissue. Do you know that? Do you know that? It is also considered as the outer boundary of the vascular tissue, or you can say that it lies in between. We can also define it in this way. It lies in between endodermis and vascular tissue. It lies in between endodermis and vascular tissue. And now we can define it in both the ways. Yes or no? We can define it in both the ways. Yes or no? Quick. Yes or no? Tell me, bache. Tell me. Quick. So, bache, pericycle in your dicot stem. In dicot stem, please make the notes. In dicot stem, this pericycle will be, it is sclerenchymatous. It is made up of sclerenchyma. Okay, it is in patches. It is made up of sclerenchyma. In dicot stem, it is what? It is basically sclerenchymatous. Right? It is basically sclerenchymatous. Clear, bache? Right? Clear, bache? And in the case of roots, of course, in the case of roots, what will you see? In the case of roots, it will be, pericycle will be parenchymatous. It is made up of parenchyma cells. Fine. It is made up of parenchyma cell, and this pericycle, in the case of dicot roots, it will also help in the formation of the secondary meristem. That we will discuss later. I'm not mentioning this point here. If you want to note it down, you guys can. Okay, so pericycle, what will it do in dicot roots? This is going to form the cambium. Okay, this is going to form the cambium. Okay, so it will form the secondary meristem there. Done? Done. And now, bache, all the tissue inner to endodermis, all the tissue inner to endodermis means your pericycle, medullary rays, or pith, right? All the tissues inner to endodermis, that is your pericycle, medullary rays, pith, they form stele. This part is known as stele. So outside the pericycle, the part is extrastellar, but all the tissue present inner side of endodermis is your, yes, what is it? It is your pericycle, it is your stele. Clear, bache? It is your stele. Clear? So it will include your, yes, it is going to include what? It will include your pericycle, vascular bundle, pith, medullary rays, everything. Okay? Done? Done, bache? So if there is any doubt in this part, do let me know. If there is any doubt in this part, do let me know. Done. So vascular bundle is also there. Vascular bundles are also there. They form the stele. So all the tissue present inner side of the endodermis, that is making the stele. That's all. Clear, bache? That's all. So this is about the ground tissue. Right, so let's have a quick revision of it. So ground tissue system is the largest tissue system. All the tissues except epidermis and vascular tissue system comes under the ground tissue system. So ground tissue system is usually differentiated, right? In monocots, you will not see such differentiation, right? In the case of monocot stem, if you will see the structure of a monocot stem, the internal structure of a monocot stem, there the ground tissue is not differentiated. You will not see the cortex, the endodermis and all there, right? But here in other cases, you are going to see this. See, in GTS of the leaves, in the case of leaves, the ground tissue system is simply mesophyll. So obviously it is photosynthetic as well. And here the GTS include cortex. In cortex you have hypodermis, you have general cortex, you have endodermis, then you have pericycle and the pith. Pith is the central part, parenchymatous cells are present there. Okay, now when it is hypodermis, hypodermis is only there in the case of stems. It is absent in the roots. General cortex will include parenchymatous cells which are meant for the storage of food. Then comes the endodermis, the innermost layer of the cortex. This is a previous year question. It is what? It is the innermost layer of the cortex. So in the roots, that endodermis is having the Casparian strips which are water permeable, which will not allow the water movement. And these, this endodermal layer, this endodermis is the starch sheath in the case of stems. This is what you need to keep in your mind. Okay, and yes, right. Okay, done, bache. So in the dicot stem you will see endodermis. In the monocot stem even the endodermis is absent. Fine. Next part to it is pericycle, which is the outer boundary of vascular tissue, and you can also define it like this, that it is present in between endodermis and vascular tissue. So in the case of dicots, it is having sclerenchymatous tissue, or sometimes it is mixed with the parenchyma also. But in roots, this pericycle is made up of parenchymatous tissue, and this pericycle in the case of dicot root will make the cambium. This is what you have to remember. And all the tissue present inner side to the endodermis, pericycle, vascular bundle, your medullary rays, pith, they are forming the stele. Okay, they are forming the stele. So pith is nothing, central part, it is made up of parenchymatous cell. Okay, it is a central part made up of parenchymatous cell. Next is the vascular tissue system, and of course a very important topic. Okay, it's a very important topic. Next is what? Vascular tissue system Here you are going to talk about the vascular bundle. That includes your xylem and the phloem. Any doubt here? That will include your xylem and the phloem. So can I say that vascular tissue is present in the stellar region? Might be you will find this word. Stellar region. It is the part of steel, na? It is the part of steel, na? So can I say that vascular bundles are present in stellar region? Of course. So the part which is present outside the endodermis, that is extrastellar region. Steel word we use for pericycle, vascular bundle, and for the pith. Okay, so yes, we can say that vascular tissue is present in the stellar system, right, Bache Shiva Kumar? It is present in the stellar system. Can we say so? Yes or no? Yes or no? Okay, so vascular tissue system mein you will study about the xylem. You know that the function of xylem is to conduct water and minerals, phloem, and then it comes the cambium, right? Then it comes the cambium. So, bache, remember fascicular cambium, intrafascicular cambium. Now we will discuss. So, xylem, phloem, and cambium is the major part there. Now, bache, when you talk about the vascular bundles, there are two things. One is radial vascular bundle. Second thing that we need to discuss is conjoined collateral vascular bundle. Radial vascular bundle, conjoined collateral vascular bundle. Now, radial, here, xylem and phloem are present at different radii. Like this is the arrangement. In this type of vascular bundle, your xylem and phloem, they are present at different radii. How are they present? They are present at different radii, means they are present at different radius. Let me show you. Okay. See, look at it. Students, please observe it carefully. Now you have to answer a few questions, okay? Uh-oh. Once again, huh? I'm telling you, if you will master this portion, then there will be nothing difficult. That's the point here. See, radial vascular bundle. What I just said. See, R for radial, R for root. That's how you have to remember. R for radial, R for root. Everyone just type it in the chat section. R for radial, R for root. है ना? R for radial, R for root. So radial vascular bundle is present in the root. This is what we understood. Now when you look at it, see, I told you xylem and phloem are present at different radii. This is a type of arrangement where xylem and phloem is present at different radii. This is the radius of xylem. This is the phloem. So they are present at different radii, yes or no? This part, red colored xylem, green colored is phloem. Red colored is xylem, green colored is phloem. Is it clear? Red colored is xylem, bache. Green colored is phloem. Tell me any doubt here. Quick. Yes. Quick. Tell me. They are present at different radii. And bache, why am I using the color red or green? Why am I using the color red for the xylem and the green for the phloem? Because after double staining with safranin and fast stain, this is extra, okay? After the double staining with safranin, with safranin and fast green. Okay, so you will see your xylem as red and phloem as green. That is why I'm using these two colors to denote xylem and phloem. Okay, so after double staining with safranin and the fast green, right, whenever we do the section cutting, whenever we color that section to see it under the microscope, after double staining, Dina, after double staining with safranin and fast green, the xylem will show the red color, phloem will show the green color. So can you see it is present at different radii? Yes, students, can you see that they are present at different radii? So it is the radial vascular bundle. Got it? It is the radial vascular bundle. Now, here, in between these, okay, in between these xylem and phloem, can you see this portion which I'm, yes? Can you see this portion here? I'm using the pink color for this. I'm using the pink color for this. The central part is the pith, of course. The central part is the pith. Here, in between these xylem and phloem, this part is what? It is the parenchymatous tissue. You also use the word conjunctive tissue for it, right? It is the living tissue. We also use the word conjunctive tissue for it. So R for root, R for radial, clear? R for root, R for radial, clear? And bache, when I was drawing the xylem, so this is how I was making it. Yes, this is how I was making it. Clear? That protoxylem is facing periphery, metaxylem is facing center. So what type of arrangement it is? What type of arrangement it is? What type of arrangement it is? Yes, quick. What type of arrangement it is? Quick. Anyone in the class? Protoxylem is facing the periphery, metaxylem is facing the center. What type of arrangement it is? Exactly, it is exact. I don't know why you are taking so long. So in the case of roots, the vascular bundle will be exact or radial. Xylem arrangement is exact, vascular bundle there is radial. Right, vascular bundle there is radial. Now next is conjoined collateral, and here you will see two type of vascular bundle, open or closed. Fine. Here you will see two type of vascular bundle, open or closed. Let me show you. Okay, conjoined collateral. Conjoined collateral. So you will find such vascular bundle in the case of stems. So bache, hits conjoint and collateral. Xylem and phloem are present on same radius, or radii for plural, radius for singular. They are present on same radius. Fine. Fine, bache. They are present on same radius. Now, conjoined means they are attached. Right? They are present on same radius. Collateral, because they are present on lateral sides. Here we have two categories. Let me show you. Closed vascular bundle and open. It is closed and open type of vascular bundle. It is closed and open type of vascular bundle. Right? What is it? It is closed and open type of vascular bundle. Okay? Now see. Bache, this is how I remember. Vascular bundle is open. It is open for secondary growth. This is my trick to remember this. It is open for secondary growth. When I'm saying vascular bundle is open, it means it is open for secondary growth. It means it is allowing the secondary growth. Secondary growth is possible in this case. So when it is the open vascular bundle, cambium is present. Okay, cambium is present. What is present here? Cambium is present here. Now let me draw this. This is actually the open and the closed vascular bundle. What is it? It is the open and the closed vascular bundle. This one is open. Okay, this one is open type of vascular bundle. So here, bache, you can see this one is closed type of vascular bundle. Okay, it is, see, conjoined collateral. Why conjoined and collateral? Because xylem and phloem, they are present on same radius, isn't it? Xylem and phloem, they are present on same radius. So it is conjoined collateral, right? They are attached. They are attached together. They are present on same radius. That is why we are saying it is conjoined, and they are present on the sides like this, right? They are present on the sides like this, on the lateral side. So that is why we are using the word conjoined collateral. Now, when I'm saying it is closed vascular bundle, when I'm saying that it is a closed vascular bundle, means no cambium, present within. So obviously no secondary growth is possible. So such type of arrangement you will see in monocot stems. Okay, and now here the cambium is present. Which cambium it is, bache? It is your intrafascicular cambium, which is also known as fascicular cambium. It is your intrafascicular cambium, which is also known as fascicular cambium. What is it? Intrafascicular cambium, also known as fascicular cambium. Okay? So because here cambium is present, so it's open type of vascular bundle, right? It will be there in the case of your dicot stems, and here the secondary growth is possible, right? Here the secondary growth is possible. So this cambium is intrafascicular cambium, also known as fascicular cambium. It is the intrafascicular cambium, also known as fascicular cambium. Clear? Clear? Sure, bache? Sure? Are you sure? So it is conjoined and collateral, yes or no? It is conjoined and collateral, yes or no? है ना? So you will see such type of arrangement in the stem. So if it is open, that type of arrangement will be there in the dicot stem and gymnosperms. If it is closed, there is no cambium present there. Okay, there is no cambium present there. So closed type is there in monocot stem and leaves, and even in the leaves. So open type is there in dicot stem and gymnosperms, and even in the gymnosperm stem. Fine, even in the gymnosperm stem. Any doubt? Any doubt? Sure? Sure. So here, bache, if you will see, this protoxylem is towards the center. So here you will see the xylem is endark. What is the arrangement of xylem here? What is the arrangement of xylem here? Xylem arrangement is endark. It is facing the center, right? It is facing the center. Okay, it is facing the center. Is it clear? Yes, is it clear? So, bache, we have one more type of vascular bundle. That is your bicollateral type of vascular bundle. Right? Here you will see xylem and phloem. This is basically the cambium. So, that type of arrangement is bicollateral vascular bundle. That type of arrangement is bicollateral vascular bundle. Clear, bache? Right? It is bicollateral vascular bundle. Okay? Okay? So it is bicollateral. Done? Fine. So that type of vascular bundles are there in the stem of Cucurbitaceae family. Cucurbitaceae, A-C-E-A-E. In the Cucurbitaceae family and in some members of Solanaceae, only outer cambium is functional. Fine. Here you will see only outer cambium is functional, that's all. So sometimes it can be bicollateral open conjoint type of vascular bundle. Okay. It is bicollateral open conjoint type of vascular bundle. And sometimes, you know what happened, bache? Your xylem is there in the center and phloem is covering it. Right? The phloem is covering it. So sometimes this type of vascular bundle is also there, concentric type of vascular bundle. So this arrangement is amphibasal, right? Why is it amphibasal? Because xylem is there in the center and the phloem is there in the surrounding. Okay, phloem is there in the surrounding. But I am not preparing for CUET, Sumaya, and it is not a doubt, it is a useless question. Okay, so that is all about the vascular bundles, and that you people should remember, that you people should focus as well, right, bache? So this is all about the tissue system. Okay, this is all about the tissue system. So guys, next part, and yes, here you guys can see. So just have a look. Radial arrangement. So R for radial, R for roots, and how xylem is present there? Exarch. This is the closed conjoined collateral vascular bundle. So no secondary growth. No secondary growth. You will see such case in the monopod stem and the leaves. Here it is open conjoined collateral vascular bundle. Okay, so you will see the cambium here. You will see the cambium here. In dicot stem and gymnosperm stems, we are going to find it. Okay, so that's it. That's all about the tissue systems. Next part is of the sections. So earlier I was thinking na bache ki I will do it in the next class, but I think we can do it. Let's revise it in this class only. Next class may we will directly start from the secondary growth in stem. Right now we are in that flow na. Hai na? We are in that flow. Let's finish it. What say? Let's finish it. And trust me, it's very easy. Trust me, it's very, very, very easy. Because you already know everything. Chalo, theek hai, it's okay. We will discuss it in next class. Okay, I'm leaving you today. We will discuss it in next class. Till then, you can revise the tissue system, bache, and then it will be easy for you people to, yes. Arey. Just a minute. Okay, so next class will be there on Sunday at 1 p.m. and I was thinking that I will start with the secondary growth only, but it's okay. We will discuss the sections there. Sections will hardly take your half an hour if you have revised this epidermal tissue, the ground tissue and the vascular tissue, because this is literally easy. Okay, this part is actually very easy. So see, I have already told you where will you find the proper arrangement, proper division of GTS, right? And where GTS is mesophyll, all the special characters also I have mentioned, like Collenchyma is present in dicot stem, Sclerenchyma is present in monopod stem. In roots you will not see such hypodermis and all. So if you will revise it properly, if you will make the notes, then trust me, this chapter is very easy for you. This chapter is actually easy for you then. But it's just that, bache, you are not consistent. You do not pay attention, and that's why you lag always. So tomorrow you will get one video on how to read the NCERT. Please check that video, right? Please check that video. It will clear all your doubts. Fine, and you will learn how to make the notes as well, and which part you should focus more, and how to read the NCERT of physics and chemistry as well. Clear? And moreover, bache, I'm telling you this thing again, the batch which is started by our HSP sir is amazing. You can see here in the description box, right? You can see here in the description box, you will get the link. Just click on the link, bacho, and join this batch, right? Earlier you used to pay lakhs for such good teachers. Now you are just getting this particular batch at a price of 1999, literally. Okay, and HSP sir is very good in the chemistry. I know it very well. Even I have seen his lectures, and you know now for your NEET also, even I will prepare one unit. So I'm going to watch HSP sir's class. Maybe I'll see Wasim sir's class as well, but HSP sir ki class I am going to watch. Okay. Done. So see, this batch is at a price of only 1999. Just. And here in this batch, what is the wonderful part? The sir will teach with the help of mind maps. And you know that, right? He is wonderful in it. Okay, so join it. Take care. So I'll meet you on Sunday, and do check the video. And I will share the PDF in my Telegram group, that is Love Biology by Ambika. So take care, stay tuned, do share the link with others, and you know what you have to do. It will hardly take your 10 seconds. Do mention in the comment section you like the session or not. Okay, you like the session or not, please do let me know. Yeah, thermodynamics and electrostatics, Shiva Kumar, I will start it. For sure, I will start it. Okay, so take care. Thank you so much.